Hello everyone, welcome back to my channel. My name is LEE ZHI XUAN. This video is the free class video for STPM Biology Semester 2 Chapter 10 Control and Regulation, special designed to those STPM candidates that take STPM Biology subject. So this is the third chapter for this semester. So this is the learning outcome based on the NPM syllabus stated. So in this chapter, there are only two main big topics, which is 10.1 nervous system and second topic hormone. And for the first big topic nervous system, we only learn about the human nervous system only, because plant does not have a nervous system, and we also do not learn about the animal based nervous system. We only learn human nervous system. But for the second big topic, we not only learn human, but majority of the topic we will learn about plant hormones.
In this lesson, I'll be using these figures to guide our explanation. If you've already purchased my notes, you'll find the full, detailed explanations written out there to follow along. If you'd like to get your own copy of these notes, feel free to contact me directly.
So let's us go through the first topic about the nervous system. So before we go through the nervous system, of course, you need to know the basic structure of the nervous system is neuron tissues, which is neuron cells that you already learned in semester 1, chapter 3. Okay. So this is just a recap only. So basically, before you go deep about this topic, you should know the structure of the neuron and the class of the neuron. There are three types of the neuron, right? Sensory neuron, interneuron, and effector neuron. Like, okay. So this is the basic structure of the neuron. You know that in sensory neuron we have the cell body, we have the axon, along the axon, and then we have the dendrite, all there, and we have the node of Lanville and the one cell, all this. So you need to know their structure very well, and also each of the structure plays the role. Okay, so this is actually just a recap only. So I think I don't need to talk much about this. If you want to know more detail about this, please refer back to your semester 1. Okay, so this is the one of the structure of the neuron in the nervous system we call as glia cells. Glia cells. So where is it located? So normally it is located at the cytoplasm to support the neuron tissue and play the role in insulating the axon of the neuron and regulate the extracellular fluid surrounding the neuron. So you can see here, see this diagram. So normally the glia cell is located at here located at here. It is to support all the nervous system, neuron cell in the nervous system. So actually this one, you don't need to know more detail about this. You just want to know they have the presence of the glia cell at the nervous system and its just major function, because this one actually is the basic structure of the nervous system that you already learned in semester 1. Okay, so next we go through the basic nervous system. So actually this topic you also already learned in SPM, just a recap revision. So we know that in nervous system we have two types of the nervous system, which is the CNS, central nervous system, and PNS is the peripheral nervous system. So these two types of the nervous system play the different role in the control and regulation in the human body. So normally in central nervous system, including the brain, highly developed brain that is continuous with the dorsal and including the spinal cord, and this organ normally is integrated to incoming information and determine appropriate response. So in general, central nervous system is normally located at the brain and your spinal cord. Brain and spinal cord is including in your central nervous system. So normally its function is to receive the information, integrate the information, and integrate this kind of information and determine what kind of response to the peripheral nervous system. So normally peripheral nervous system is to make up of the sensory receptor and the nerve, which are the communication line for the central nervous system. So in this diagram, you can see here, normally when you have when peripheral nervous system that containing afferent neuron, when it have the receptor is detected something else, it will send through the afferent neuron, and this information will go to your brain. So this brain is your central nervous system. It will including the central nervous system, and this our brain will integrate your process all this kind of information, and it will send the desired action to the receptor, which is including in peripheral nervous system. So it will including send the receptor to the send the impulse to the receptor through the sensory neuron, efferent neuron, and by either through sympathetic nerve or parasympathetic nerve to let the effector to do the action. Okay, so this is the simple one. This is just a simple one. So normally, is the central nervous system is the key component, like the key robot in your brain. Not robot. It's a very important component that can integrate the information to either you want to do or not. So for example, when you have received the information about the pen, so about the pen. So what should your hand to do? Maybe your hand, maybe you already touched the something which is very hot, and then your hand normally automatically will go will away from the hot things, right? So this is what the nervous system do lah. So this is a basic central nervous system and peripheral nervous system work. So this is a summary signal pathway of the nervous system. So normally E is the reception by the sensory receptor in the skin. Your skin have the receptor. It will detected the signal, and if this signal will send through the sensory neuron to the central nervous system, and this central nervous system will integrate your information. So for example, one of the example is the gaseous exchange. You already learned in semester chapter 1. Chapter 8, gaseous exchange. Right, breathing mechanism. What is the receptor of the breathing mechanism? You know that it is a peripheral chemoreceptor, right? They're located in aortic and carotid body. What it detect? It detected normally is the your carbon dioxide concentration change, your pH change in the blood. When it detected, it was sent through the central nervous system. So in breathing mechanism, what is your central nervous system? Medulla oblongata, right, in your brain. So your medulla oblongata was integrate this information and transmit the impulse to the to your efferent, to the motor neuron. So what is the motor neuron of that breathing mechanism? Vagus nerve and another neuron cell. I already forget. We got two type of neuron that transmit the signal one. So this is a basic mechanism, and it will go through your effector. So in breathing mechanism, your effector is lung, including intercostal muscle, rib cage, diaphragm. All that is your effector, and your effector, your muscle either contract or relax. So normally it's contract. Yeah. So this is a summary signal pathway of the nervous system, and this signal normally is go through along the semester. Not only breathing mechanism, especially in homeostasis. Yeah, in homeostasis, there are many topic is relating about this. So you need to know the relationship. Okay, so this is another diagram about the central nervous system and peripheral nervous system. So normally when you detect the receptor, your peripheral system will be transmit your information to the central nervous system, and the central nervous system will integrate the information and transfer back to the peripheral nervous system, and your peripheral nervous system will detect the signal from the central nervous system, either it is through the sympathetic or parasympathetic one. Right. So what is sympathetic and parasympathetic? Later we will go through it. So this is overall of the process. Okay, so next we go through sympathetic and parasympathetic. So if you go through the MPS syllabus, it actually is at the last part of the topic. It is not the initial part, but for me, I think it will be very easier directly to you when you already learn the nervous system. Directly, you learn about the sympathetic and parasympathetic is very easy lah. So normally in the peripheral nervous system, or we call as autonomic nervous system, you have two type of the division, which we call as sympathetic or parasympathetic. Okay, so what are these two neuron different? Okay, so normally you can see here when in sympathetic nervous system, you can see here your pre-ganglionic neuron and post-ganglionic neuron are different in length. You can see here sympathetic, your pre-ganglionic neuron is shorter than post-ganglionic neuron. But for parasympathetic, your pre-ganglionic neuron is longer than post-ganglionic neuron. And the acetylcholine, the neurotransmitter that involved in sympathetic is acetylcholine and acetylcholine for sympathetic ganglia at the ganglia part. Both of them are involved in acetylcholine, but the difference is at the efferent part. Post-ganglionic neuron at the effector part. So for the sympathetic, your neurotransmitter is noradrenaline. For parasympathetic, your neurotransmitter is similar as the ganglia one, is still acetylcholine. Okay, so these two actually got two different function, because these two neurotransmitter play the role in different way for the muscle to do something. So later we will see the role of the neurotransmitter for each of the different type of neurotransmitter. So if you still remember in last chapter, chapter 9, in the regulation of heartbeat, you know that acetylcholine will decrease your heartbeat rate. For noradrenaline will increase your heartbeat rate. That kind of mechanism. Okay, so in this chapter, we will learn more detail how these two neurotransmitters plays this kind of role. So this is just a summary of the parasympathetic and sympathetic division for each of the effector action. So in general role, parasympathetic play the role in rest and digest condition. So in simple, it's relaxed one. When the parasympathetic division is stimulated, it send the signal to your effector. Normally, it's during your rest, is very relaxed, or digest condition. But for sympathetic, it is the way when sympathetic is stimulated, meaning that effector is responding to danger and stress condition. So you can see here. You can see here. You see this kind of diagram. So for example, like heart. For parasympathetic, it slow the heartbeat rate. Your cardiac muscle contraction will be reduced. But for sympathetic, it will increase your heartbeat rate. Why? Because it is responding in dangerous way. When you are very dangerous, highly panic condition, very stress condition, your heartbeat is very fast, right? Compared to relaxed condition. So similarly about this, and also digest condition. So that's why it stimulates activity of pancreas, right? We know that pancreas can secrete hormone, insulin, glucagon, all that. All that because it including in digestion, is similar to the gallbladder. Gallbladder also including in the nutrient digestion part. So all this. So for this, you need to know all about this, because in examination so far for now, structural question and essay question in previously so far not come out yet, but it will come out in objective question. So normally objective question, they just come out in table format and then just write parasympathetic and sympathetic, and then all all these kind of factor just say parasympathetic division, dilate pupil of eye, is it correct or not? So you should know all these effector. So in simple word, you just imagine in rest and digest condition, only parasympathetic work. And you just imagine when your eyes is in rest and digest part, what your eyes do? What is your salivary gland do? What is your lungs will do? Heart will do? And pancreas will do? So it is just a very easy one, not very hard. Just very general. You just can imagine yourself When you are very relaxed, what you will do, what your organ supposed to do. Okay. Okay. Now, that's all about the nervous system in human. Okay, that's all about the basic nervous system. Next, we will go to the very new topic that you never heard it before in SPM, which is resting potential. Okay, before I go through this kind of topic, I just want to tell you, we know that in nervous system, the signal is transmitted along the neuron based on electrical impulse. Okay, can you imagine that? How the electrical impulse is transmitted from here to here? Have you think about that before? Because in SPM you just know, oh, electrical impulse transmitted along the way. But how does this electrical transmitted along? And we know that between the neuron, they have the interchange, right? Have synapse. In SPM you also learn has synapse. But synapse has the synapse, and neurotransmitter play the role to transmit this impulse from one neuron to another neuron, right? But how does it transport? How this electrical impulse can be transported? And along the neuron, how the electrical impulse can be transmitted from one part to another part? That is the clue one. Not like the physics wire, you just got a signal, then pass through along. No, not like that. They have the mechanism one. So the mechanism is based on the resting, scientific terms we call as resting potential and action potential. Okay, so in resting potential, the definition is that when the neuron is not sending any signal, it is in resting state, meaning dia ada rehat lah, dia does not sending any electrical signal at all in that neuron. So in that neuron, it is negative one. Your membrane potential or voltage, in physics we call as voltage, but in biology, because it is in the neuron membrane, so we say membrane potential is at negative one. It's about negative 70 milliwatt lah, milliwatt. So it is negative. Okay, so for now you need to know the structure of the neuron plasma membrane. Okay, in neuron plasma membrane, we know that we have sodium and potassium pump. This is the general role, sodium potassium pump. Right. So if you still remember what is the sodium potassium pump in active transport. In active transport, it is the movement of the sodium and potassium ion against the concentration gradient, right? Because it requires ATP. So in normal situation, when your your sodium potassium pump will pump out 3 sodium ion and out of the cell and 2 potassium ion inside the cell, right? This is the general characteristic that you already know. Okay, so for now you need to know why in resting potential the membrane potential is negative inside the cell. When we talk about the resting potential, we are saying that the voltage inside the cell one, not outside the cell. Outside the cell, we don't need to care one. We just want to care about inside the cell. So meaning that in resting potential, your inside the cell is negatively charged. It is not so positively charged, it is negatively charged. And also in the plasma membrane of the neuron cell, we also have the potassium, also have the voltage gated ion channel. Okay, so this one is later I will explain it. For now, you need to know why in the resting potential inside is negatively charged. Okay, so before we go through it, we need to know the definition of membrane potential. So it is the differences in the concentration between specific ion inside the cell and those in extracellular fluid. So meaning that outside is positive, inside is negative. So that inside is negative. Okay. So there are 2 major factors affecting the mental potential. Membrane potential are difference in the concentration between specific ion and those in the extracellular fluid, and selectively permeability of the plasma membrane to this ion. Meaning that in your resting potential state, if there are no electrical signal, your potassium ion is about 10 times greater inside the cell than outside the cell. Meaning that potassium ion inside the cell is higher than outside the cell. Outside the cell. And for sodium ion, outside the cell is higher than inside the cell. So in simple word, in resting potential state, potassium ion is higher inside the cell. But for sodium ion, is higher outside the cell. So that's why it use the sodium potassium pump. Sodium potassium pump. Because sodium ion is outside. So if you want to pump out your sodium ion, you require active transport 1 because it is against the concentration gradient. Okay. So this asymmetrical distribution across the plasma membrane at rest is brought about the action of selective ion channel and ion pump. And there are 3 types of the protein channel in neuron called voltage-gated ion channel, passive ion channel, and chemically activated ion channel. Okay, so what are these? So chemically activated ion channel is the sodium potassium pump. This is the general one. Okay, passive ion channel is the core protein like this. This one. Meaning that it does not require the protein channel is always open, and the movement of the ion is based on the concentration gradient. So normally, if you including passive ion channel, meaning you already say a passive, meaning that the concentration is moved down across the concentration gradient. But there are another key channel which is voltage-gated ion channel. Okay, this channel is very important to maintain the membrane potential because in voltage-gated ion channel, in resting potential state, it is closed. It is closed. All the channel is closed one. Closed one. It will only open when they have the situation we call as action potential. So in the neuron, there are 2 main types of voltage-gated, which is sodium ion and potassium ion. And both of them are closed in the during resting state. They will only open when they have action potential. Okay, so this is a basic I need to tell you. Okay, so you can see this. Okay, so this blue color one is sodium potassium pump, normal one. And this one is the potassium channel. This is what we call as passive ion channel. Potassium channel and sodium channel. So we know that in resting state, what is happening is potassium ion inside the cell is high. Like you can see this diamond shape. Diamond shape is called representing potassium ion. The circle one is sodium ion channel, sodium ion. So you can see here inside the cell, potassium ion is higher than outside the cell. But for sodium ion, outside the cell is higher than inside the cell. Inside the cell. So the investing potential, the charge inside, outside of the axon is maintained by this sodium potassium channel and this sodium potassium pump. So it need to maintain that potassium ion always must be higher than outside one. It cannot be switched. If it is switch meaning that when the sodium ion is higher inside the cell, meaning they have the action potential already. So investing potential, it must be balanced like this. How does this balance? The passive ion channel only allow potassium ion or sodium ion allow. So if this is potassium channel, it only allow potassium ion move out from the cell. Why it is move out from the cell? Because potassium ion is higher inside the cell. So the potassium ion is moved outside the cell because down the concentration gradient, right? So similarity for sodium channel, this is vice versa, meaning that sodium ion is moved into the cell. Okay, move into the cell. Okay, so why does this because normally in resting potential, the plasma membrane is more permeable to potassium channel instead of sodium channel. So that's why that even though there are sodium ion coming into the cell, but still potassium ion still higher than outside the cell. Why? Because the potassium channel amount is higher than sodium channel. So that's why you can see here in this diagram, there are 4 potassium channel, but only 1 sodium channel here. And you don't need to forget that, and we have potassium pump, still can maintain it because sodium ion is still pumped out from the cell and potassium ion pumped into the cell. So it is a maintaining lah. So if the number of the potassium channel and sodium channel are equivalent, meaning that the ion of the potassium and sodium are balanced. We don't want balance. We need to have one unbalanced one. Potassium ion inside the cell must be higher. Sodium ion outside the cell must be higher. We need to unbalance. So the number of the channel for potassium channel is higher than sodium channel. Okay. So this has only a minor effect. However, the concentration gradient created by the pump are significant. Okay. So this, although it is can say that it is just a minor effect, but still it is important to balance it. Okay. Okay. So when you are sodium ion is outside the cell, and due to the number of the sodium channel is very low, very less, so that you want the sodium ion diffuse back into the neuron is very difficult, because sodium channel is amount is very low. However, your potassium ion is easily pumped out. You can see here, is easily to be pumped out. To be pumped out. You can see potassium ion is easily to be pumped out. So potassium ion will diffuse out of the axon, increasing the positivity charge in the extracellular fluid relative to the charge inside the cell. Okay. So maybe some of you will ask, although you say that potassium ion is still positively charged ion, so in general, in theory, it should be also positively charged, right? But you need to know, even though potassium ion channel is very high, but it is easily to be pumped out. You easily to be pumped out one. Pump out one, compared to the sodium channel. Because sodium channel want to pump it, if you want to diffuse the sodium channel into it is very difficult. But potassium ion is very easily come out from the neuron. Why? Because we have sodium potassium pump. We only pump 2 potassium ion inside the cell, everyone. But we have many potassium channel which can easily diffuse the potassium channel out from the neuron cell easily compared to sodium. So there is a different incubation of other ion and molecule across the membrane, especially negatively charged protein. So which are at a higher concentration site inside the cell than outside. Okay. So because of this, they will have the large N ion. Normally is protein or protein can be accumulate inside the cell. Because in intracellular, you need to know that inside the cell not only have potassium and sodium only, we also have the large N ion, other N ion inside the cell one. Normally it's protein like amino acid inside the cell one. So normally this amino acid is large and also negatively charged. So because the membrane is not permeable to the negative counter ion and potassium ion is diffused out, so they are remain inside the axon, meaning that this large ion cannot pass through because it is large, and there are no channel for this ion one, because it is for this channel one, because large ion cannot directly pass through the phospholipid. You need to know, only the small molecule can pass through the phospholipid. Other than that, large molecule normally you require a protein channel. Because there are no protein channel, so that all the N-ion channel, large N-ion channel is remaining inside the cell, and the potassium ion still diffuse out from the cell, of the cell. So they are remain inside the cell, inside the axon. So positively charged build up outside the membrane and a negatively charged inside the membrane. So this is the overall theory. Why inside the cell is negative, outside is positive. And this we call as resting potential. So this is the concept about the resting potential. So this table, I just want to tell you, potassium ion channel, you can see inside is high, outside is low. And sodium, inside is low, outside is high. Chloride, you can ignore it, don't need to focus, ignore it. But large ion inside the cell is very high, because it already maintained at there already. But outside does not have, because this And ion cannot easily diffuse out one because they do not have the ion channel for that. For the protein to diffuse out. So cannot, so no anion at there. So this is why that resting potential is negatively charged in the axon. So simple way, resting potential inside the cell is negatively charged, outside the cell is positively charged because there are no electrical signal inside the cell. Okay, so this is resting potential, resting state. Okay, now when they have the signal, when your receptor already receive the signal, something like chaotic body, chemoreceptor, all that already receive the signal. Okay, now your neuron was starting to transmit this kind of signal to the central nervous system, right? So how this signal form, we call as action potential. So it is an electrical signal that travels rapidly down the axon into the synaptic terminal, meaning that when the receptor at here already received the signal and this impulse was transmitted until synapse at here. Along this way, we call as action potential. When they have the electrical signal at here, we call as action potential. And you need to know in action potential, the signal is coming from one part to one part. Meaning that it is also say that, when they have a signal, then directly all part is positively charged. No, not all part is directly positively charged. It have the area one. Later we will know. But for now, you need to know how the action potential is formed. How, meaning that, in general term, how do you want to change this negatively charged in the axon inside of the cell become positively charged? Because if you want to electrical signal, you need to have positively charged. So in general, become inside the cell become positively charged, outside the cell become negatively charged. So in simple word, sodium ion inside the cell now is higher than outside the cell. Potassium ion outside the cell is higher than inside the cell. How this switching of the concentration happen in the axon during action potential. So this is the theory that you need to learn. Okay. So there are 5 main phases. First phase is resting potential, like what I said before. Second phase is depolarization phase. Depolarization. Okay, what is depolarization? Okay, never mind. We go through first. After that, also the third phase also in some of the book also say depolarization. But some books still say depolarization, but in more scientific term is rising state. Third phase, rising state. And then the fourth phase, you can see the line is dropped. This we call as falling phase. But it is recommend not to say falling, say repolarization. RE. Just now is depolarization. The fourth phase is repolarization. And then after that, you can see here, the membrane potential is even negative than the resting potential. We call as undershoot. And then after that, it back to the resting potential. Okay. So we will see it one by one. Okay. One by one. Okay. So in general, in resting potential state, you does not have any electrical signal. So inside the cell is negatively charged, outside the cell is positively charged. And beginning of the action potential, we will use the voltage-gated ion channel. So they have 2 voltage-gated ion channel, which is sodium and potassium. So they were starting to open and close. Okay, so which open first? Okay, imagine you need to see. I already said in action potential, you need to make inside the cell positively charged. So how the sodium ion outside the cell move into it? So you need to open your sodium channel, the voltage-gated sodium ion channel. In examination, you need to say voltage-gated lah. You just not say sodium channel. If you say sodium channel is wrong, because if you say sodium channel, meaning that is the normal passive ion channel only. You need to mention voltage-gated sodium ion channel. You need to open it. Okay, how make it open? Because if resting potential is closed, how do you want to make it open? So there is the depolarization. Okay, when the nerve impulse is arrived, meaning that they have the stimulus in your receptor. Okay, your receptor already receive the signal. Okay, receive the signal. Your voltage gated ion channel starting to open, but not all. At the depolarization phase, not all, some only little amount starting to open. So when it is open, what happened? Because this is it is actually function like the sodium passive ion channel one. How when it is open, meaning that the sodium ion will move down the concentration gradient. So in resting state, your sodium ion is accumulated at the outside the cell. So all the sodium ion was starting to influx into the cell, into the cell. So it was starting to depolarize the membrane. When the sodium ion is entering into the cell, it is mean that depolarization starting to occur. And if the depolarization reach the threshold level, meaning that when the membrane potential value starting to increase until reach the threshold level, it will trigger an action potential. So it will trigger rising phase. Meaning that when the sodium ion channel starting to entering into the cell, and it will accumulate means when it is accumulate, meaning the membrane potential starting to increase until it reach the threshold level. So what is the exact value of threshold level? So different type of neuron actually has different threshold level value. So in this example, they use negative 48, something like that. But in examination, you don't need to know the exact value. We just the key important thing is exceeds threshold level. When it exceeds the threshold level, it will trigger the action potential directly. It will directly trigger one. So it will cause the rising phase of the action potential, rising state. What does this mean? Mean that all the voltage gated sodium ion channel open. All are open. But you need to know, during depolarization and rising state, your potassium, the voltage gated potassium ion channel still closed. They do not open one. It's still closed. Meaning that all the potassium ions still relax, maintaining inside the cell, and they have the potassium passive ion channel, right? Some of them still diffuse out from the cell, and more majority still inside the cell. The difference is that all the sodium ions already influx into the cell. So sodium ion will in fact make the cell inside of the cell is positively charged now. In the depolarization phase, at the beginning of the depolarization, inside the cell still negative. But when it exceeds the threshold level, more sodium ion already influx into the cell, meaning that it is positively charged now. It is already positively charged. Okay, this is the signal. Okay, when it already have the signal, this signal will go through another part. So when the signal is already passed through it, now the cell need to return back to the resting state. Because the cell not always at the action state one. It only becomes action potential action state when they have the signal. Once the signal go through it, then it need to back to the action with resting potential, sorry. Okay, so how it return back? So meaning that if you want to back into resting potential, how? Meaning that sodium ion need to come out already. So you want to make your sodium ion come out, how? You need to close the voltage gated ion channel first. To prevent all the sodium ion channel outside the cell coming into inside the cell already. You need to close it. So now we go through the repolarization. Repolarization, falling phase. In Campbell they use falling phase, but in real examination, it is better you use repolarization instead of using falling phase. Repolarization. Okay. Repolarization occur. So how it occur? So first of all, your voltage gated sodium ion channel close, starting close. So no sodium ion channel can enter into the axon now. So how? So and then potassium ion channel, your voltage gated potassium ion channel open now. So it's open, starting make the potassium ion inside the cell at flux outflow to the outside the cell. Make the cell inside of the cell negative again. Again. Get again. Outflow. outflow. And then after that, we go through the undershoot. Okay, you can see here, when it is in the undershoot level, the membrane potential is even lower than the resting state. So in undershoot level or undershoot level, what is happening is that voltage gated sodium ion channel still closed, but potassium still open, and potassium still open. So it make all the remaining potassium ion efflux out of the cell. As this potassium ion channel close and the sodium become unblocked, the membrane return to its resting state. And this is the process we call as after hyperpolarization. And normally in undershoot, your potassium ion permeability, potassium ion to the membrane permeability is very high compared to the resting state. So that's why that your very higher amount of the potassium ion is efflux out from the cell, making it more negative value. Okay, so that's all about the action potential. So after when the potassium voltage ion potassium gated channel already closed, then it will back to the resting state. So that's all about the cycle, one of the cycle about the action potential. So E is actually the cycle one. Once the signal already transmitted, it will go through the repolarization, your sodium ion channel closed, potassium open, and all the potassium ions starting to efflux to maintain the negatively charged ion at here. So that's all about this. Okay, so now we need to know transmission of action potential along the axon. Okay, so you can see this diagram more clearly. As I mentioned, in axon, the electrical signal is not say that when it pass through all along the axon is becoming action potential, becoming positively charged. No, it is triggered region by region. So for example, like this. When this is at the beginning, imagine this is a receptor. When it receives the stimulus comes through the first region first. And then after that, it was transmitted to the second region. Meaning that at first region, the behind part, the following part is still negatively charged. They still resting state. But only the first part is action state. After that, it will transmit to the second region and then go through third region, fourth region, and fifth region. It is transmitted one region by region. Not say that, or when they have detected that along all the axon become positively charged. No. You will say like that is no. So that's why we need to learn this. Okay. So how the action potential from here, first region, affect the second region? How it affect to second region, how it affect to third region, fourth region, fifth region until it reach the synapse. Okay. So when the action potential is initiated, your sodium ion inflow, right? Sodium ion starting to the depolarization starting to occur. So your sodium ion starting to influx into the cell. It will create an electrical current or localized current to depolarize the neighboring region of the action potential. So once there already sodium ion channel and inside are coming to the first region, first region, it was starting to depolarize the second region. Why? Because it create an electrical current from here. That can affect the second region one. So normally, during the depolarization, your affected area of the membrane is more positive to the ejection region where the membrane still at resting. Of course, right. It is at the first part is positive. So for next part, second region still negative. So this difference create the localized current. Electrical current, because of this difference, it create the electrical current. Okay. So when the least electrical current will cause the depolarization of the beginning region of the axon membrane, spread the neighboring part of the membrane, and the neighboring part of the membrane of action potential is initiated by the depolarization of the beginning region of the action potential. Okay, so when the depolarization of the first part is already occur, the neighboring part of the membrane was starting to initiate the action potential. And to left of this region, the membrane is repolarizing as potassium outflow. Okay. At the first region, repolarization, depolarization occur. And then after that, it will cause depolarization. Right? Meaning that potassium ion starting to outflow from the cell. When the depolarization of the first region start, next the second region will start action potential. Potential. So that's why it say that the neighboring region of the action potential is initiated by the depolarization of the first region. It's very simple. When they have when the first region starting depolarization, then next region starting repolarization. Okay, and then after that, it will trigger the rising state, so the ions enter, and then it will trigger repolarization again. So this repolarization will trigger another third region of the action potential. So this process is repeated again and again. That is why it kind as depolarization and repolarization process is repeated in the next region of the membrane, and the Doppler curve across the plasma membrane is cause the action potential to be propagated along the length of the axon. So this is how the electrical signal pass through. So instead of say that when the receptor receive the stimulus all along the way, directly becoming action potential, action state, directly becoming positively charged. No such thing. Please don't use that terms. Because sometimes or in SPM maybe you imagine like this, but in real actually not. So it is like this. Like this. So in the first region, okay, action potential already occur. Once it reach the depolarization, it will initiate the neighboring region of action potential, and then it will repeat again and again. So that's all about the axon, about the nerve impulse transmitted along the axon. Okay, so if you want to transmit the nerve impulse, your axon inside the cell must be positively charged so that it can be transmitted. If it is negatively charged, it is resting state. Once it being positively charged, meaning is action potential is formed. Okay, so how to make the action potential form? You need to have sodium ion inside the cell. Or sodium ion inside the cell. So that's all about the transmission. Okay. Next is about the characteristic. Okay, nerve impulse characteristic. Okay, so mostly you need to know about the refractory period. Okay, what is this refractory period? So you can see this diagram. They have two types of refractory period: absolute refractory period and relative refractory period. So absolute is between high depolarization phase, rising state, depolarization, rising state, and also the repolarization. And relative one is at the hyperpolarization, which is understood state. So what is this? What is refractory period called? So normally when you want to sodium channel, we make it activated during the falling phase and the early part of the undershoot. So E is the phase normally is your sodium channel starting to remain activated during falling phase and the early part of the undershoot at here. So if a second depolarizing stimulus occur during this period, it will be unable to trigger the action potential. Okay, so refractory period is the period for the nerve impulse to relax first. Meaning that when during this phase repolarization, when they have stimulus occur, they still no action potential. Because it require complete one cycle first. Only when the region, meaning that when this region at first region they still complete already have the action potential, and during the depolarization phase at here, when there are stimulus still happen, no action potential at here. It will not say, oh, or because got one impulse at here there. So I change a bit. So I skip up the depolarization phase, skip up the undershoot. I directly go through depolarization phase. No such thing. There are no such thing. If you want to trigger another type of action potential at this region, you need to make this region becoming resting state first. Then the stimulus come, it will become action potential again. So you need to complete this one cycle, then you can trigger another action potential. You cannot say that when doing depolarization, repolarization, or even hyperpolarization state, when they have another stimulus, no action potential occur. So this is what we call as refractory period. So the downtime when a second action potential cannot be initiated is called the refractory period. So that's why I say that in refractory period is along at here. So there are two types of the refractory period period. So absolute refractory period, so it cannot transmit another action potential no matter how great a stimulus is applied, because the voltage ion channel are already starting to inactivated. Until the gate their gate are raised, they cannot open again. Because at here still open, right? Depolarization phase open. But until it reach the repolarization phase is starting to inactivated. It already closed. Close. Once it is already closed, starting to close, but how strong your stimulus is, they still cannot open. They need to complete first. This is what we call as absolute refractory period. When enough sodium ion channel are raised, totally already closed, the neuron will enter relative refractory period and axon can transmit the impulse, but the threshold is higher, even with the limit imposed by their refractory period. Okay, when it reach here, if you want to trigger the action potential, yes, you can. But threshold level is much higher compared to here. Meaning that at here, your threshold level is not negative 55 anymore. Maybe it's negative 50, negative 45. It will up. It will increase the threshold level. Only the membrane potential reach this much higher threshold level, then it trigger action potential. If not, it will still maintaining resting potential. Then during resting potential have received the stimulus, then it repeat again. So this is the meaning of the absolute and refractory period. So why this period is important? Why must like this? Why cannot say that during repolarization I got stimulus, still already action? I trigger action potential. Why cannot? Why our human need to have this kind of mechanism to inhibit the stimulus of the stimulus? Why? So it allow the action potential is moved in one direction only. One direction only. If during the depolarization phase, starting here, got stimulus, you still trigger action potential, meaning that maybe the direction will be changed already. Because your sodium ion channel concentration between the inside outside the cell is already different. Maybe it will go backward. If go backward, it is something problem. And it also limit the frequency of action potential when successive. It limit. They don't want to make your cell is too hyperactive. If your cell is too hyperactive, meaning that your membrane potential will be increase. Because it is milliwatt, If it does not stop it maybe go to 1 watt. 1 watt meaning that your cell is electrical already. Because milliwatt still normally is still for the real situation, real life is still normal. You do not have a feeling one. If milliwatt is applied into your body, but if 1 watt, 2 watt applied in your body, you will feel something problem right here. So it need to limit. Okay, so this is what we call as refractory period. Okay, another one is all or none law. Okay, this we will focus about the threshold level. As I mentioned before, if you want to trigger action potential, your membrane potential during the initial depolarization phase must reach the threshold level. If not, then no action potential occur at here. At here? Meaning you can see this diagram, the depolarization occur at here, starting to occur, but it does not reach threshold level. The stimulus is not strong enough. So even they have the depolarization, at here, but no action potential because the stimulant is too weak. So it is either all or none law. If you have you reach the threshold potential, no matter your stimulus is too high, that is very high or low, your action potential is still the same. Not say that your depolarizing power is very strong, your action potential will be very high. No, no such thing. So that's why it's only say all or none law. Even though it just reach threshold, then it trigger. Even though the depolarizing powers suddenly increase up to zero, they still trigger action potential like this. So that's why it say all or none law. So this is one type of the characteristic. Okay. Next is the speed of the conduction of nerve impulse. Okay, so in general, if you want to increase the speed of the axon, nerve impulse along the axon, meaning that if you want to increase from here to here, if you see here, maybe need 0.5 second. Too long, I don't want. I only want 0.1 second from here to here. Can or not? Can. Based on this few type of characteristic. One of the factor is diameter of axon. Okay, so how diameter of axon? So normally, the larger the diameter, the faster the speed of the conduction. Because the smaller the diameter, the greater the resistance of the exoplasm and lower the speed of the action potential. So meaning that your diameter of the axon must be very high, must be large. If larger, then speed conduction is more higher. That's all. For temperature, of course, you need to have the higher temperature, but not too high. If too high temperature, then your cell will die. But if you increase the temperature, it will increase the kinetic energy of the ion. So of course, you can make the diffusion of the ion move more faster compared to the low temperature part. But if too high, it will be denature, your cell will die. And then your system fail. So temperature is one of the reasons. And another one is presence of the myelin shift. Okay, what is this myelin shift? Okay, so you still remember about the structure of the neuron you already learned in semester 1. You see this diagram, they have the yellow color part one. Yellow color. This one is we call as myelin shift structure of the neuron. Not all the neuron have the myelin shift, but if they have the myelin shift, it can speed up the neural the electrical impulse along the axon. So how this can transmitted? Okay, so myelin shift normally there is made up of the cell we call as the Schwann cell, and it is insulator, meaning that the electrical cannot pass through into it. It is poor conductor, poor electrical conductor. It is a good insulator. So they between the myelin sheet, between the cell, the Schwann cell, they have the gap, right? You can see this gap we call as knot of Witteville. Knot of the Witteville. Okay, this what does this cause? How this myelin structure of the axon can pass through very fast because the electrical impulse is jump from one node of Witteville to another one of Witteville. They will not pass through the Schwann cell one. The action potential will propagate more rapidly because time-consuming opening and closing of the ion channel are short. And so this propagating mechanism is we call as salutary conduction. Salutary conduction because the action potential is appear jump from one node of Witteville to another node of the axon. So this one will speed more faster. If they have non-myelinated, meaning that your action potential need to pass through one region to another region. But if let's say this part is myelinated, meaning the action potential from first region can directly jump into another the final region already. They don't need to pass through here. Meaning that all this is ignore, it will ignore. Action potential will directly pass from here. So this is the one of the factor. So there are three major factors that can influence the conduction or speed of the electrical impulse. Okay, so okay, next we go through the synapse. Okay, this is you already learned in SPM lah. What is the synapse? So synapse is the gap junction between the two neurons. So maybe it's afferent neuron and interneuron, or afferent neuron in efferent neuron lah, when they have the gap junction between the two neurons as well as the receptor and sensory neuron. Sensory neuron. Okay. Okay, so this is the basic structure. We know that at here, when the electrical impulse is coming through, we call as presynaptic neuron. And the neuron that receive the electrical impulse is called as postsynaptic neuron. And we know that in synapse we containing mitochondria that provide energy, axon terminal, and the gap here is we call as synaptic cleft. And in the end part of the presynaptic neuron, they have the synaptic vesicle that containing the neurotransmitter. So this is a basic structure of the synapse that you already learned in SPM. So I just make a revision for you. Okay, so how the impulse transmitted in synapse. So I think this one you also learn in SPM, but I think SPM is just a go through, not very detailed. Okay. When the electrical impulse is already coming through the presynaptic membrane of the axon, starting reach here, when the action, or we can say that when the action potential is arrived, it was starting to depolarize your presynaptic membrane. Okay. What happened to your when your presynaptic membrane is depolarized? Your calcium ion channel open. Voltage gated calcium ion channel open. When it open, your calcium ion starting influx into the membrane, into the presynaptic membrane. Not into the presynaptic membrane, presynaptic neuron inside the cytoplasm. So this calcium ion will cause the synaptic vesicle that containing neurotransmitter fuse to the presynaptic membrane. So it is like the activator channel, activator ion like that lah. It make the synaptic vesicle fuse to the presynaptic membrane and release the neurotransmitter. Then this neurotransmitter will pass through the synaptic cleft and bind to the receptor that located at the postsynaptic membrane. It will bind to the receptor, any receptor like acetylcholine, it will have the acetylcholine receptor. Noradrenaline, it will have the noradrenaline receptor. And this neurotransmitter will diffuse across the bind to the receptor, and the binding will cause the ligand-gated ion channel open or close to allow the particular ion to diffuse across the postsynaptic membrane, result in graded potential. Okay, so this one is need to depends what type of the neurotransmitter lah. So for example, you see here when the neurotransmitter is already bind to this receptor, it will trigger the opening of the channel, allowing the potassium and sodium pump to be activated lah. If does not have this neurotransmitter, the sodium potassium pump is not activated one. It still maintained at that one. But when they have the sodium potassium pump neurotransmitter, then it will activate it. Okay, so this one, what I need to relate to parasympathetic and sympathetic here. Acetylcholine. When acetylcholine bind to it, what happened? So this one is that you need to relate. After the ion channel is activated, it will trigger the depolarization of the postsynaptic membrane. Membrane, and when this depolarization was starting reach the threshold level, it will trigger the action potential in the postsynaptic membrane, and it will transmit the nerve impulse to the effector, to the end of the axon near the effector. So this is the synapse. So binding of the neurotransmitter into the receptor is based on the situation. For this situation is the activating the sodium potassium pump. Yeah. If does not have this neurotransmitter, this sodium potassium pump is inactivated one. But it is still once binding it is activated, it will trigger the threshold level and triggering the action potential. Okay. So this is the termination at okay. Okay, so termination of the neurotransmitter signaling. So this I based on the reference book to let you see what is happening lah. What is happening when the neurotransmitter is released, it bind to the receptor, and then what happening after next. So after the EPSP is depolarized the postsynaptic membrane and already trigger the action potential, the neurotransmitter at the synaptic cleft need to be clear. Because you need to know that neurotransmitter is remaining at day one. They cannot go back. They cannot go back easily one. Not simple say that you want to go another way, because here is the cytoplasm region one, synaptic cleft is in the cytoplasm region one. Not cytoplasm, at the extracellular fluid region, at the outside one. So this neurotransmitter need to clear. If this is not clear, meaning that this neurotransmitter can always bind to the receptor one. Maybe you go to another pair, go to another part, another region, bind to another receptor, cannot. So this neurotransmitter need to clear. So how this neurotransmitter can be clear? Either using enzyme, maybe some of the enzyme can be used to inactivate the neurotransmitter. Maybe it will break down the neurotransmitter, or catalyze the reaction of the neurotransmitter. For this, another one is reuptake, recycling. They have the neurotransmitter. Once it already bind to receptor, then this receptor will go back to the this neurotransmitter will go back to the presynaptic membrane, transport through the diffuse into the presynaptic membrane through the neurotransmitter transport channel. So this is the two major pathway how the neurotransmitter is clear. Okay. So normally when as I mentioned, when you activate the receptor is activated, it will send the impulse to the postsynaptic neuron. So how this impulse we known as two main type of the impulse is either is excitatory or inhibitory. So excitatory excitatory meaning that they have the action potential, the membrane potential change in bring the neuron closer to firing, meaning that it will trigger the action potential. For inhibit, because you already said inhibit, it will stop the action potential, no action potential. So in here, meaning that they have two kind of reaction. Once the neurotransmitter bind to it, either it stop trigger action potential or it inhibit action potential. They have two major ones. So in this diagram is a summary of the postsynaptic potential. So when you have when in postsynaptic membrane, when you receive the stimulus receptor from the synapse, if the action potential, your membrane potential does not reach the threshold level, then your postsynaptic membrane does not have any action potential. But when you have the temporal summation, meaning that you add it, you will trigger the action potential. Okay, what does this mean? You can see this diagram. When you receive the action potential from E1 at here, E1, and there are another here E1, but E2 does not have received any signal. E2 don't have any signal, only E1. So the first E1 signal very low. Okay, after a few period, you receive another signal E1 again, same direction. but still the threshold it does not reach the threshold level. So unfortunately, this postsynaptic neuron does not trigger any action potential, meaning that even though in the sensory neuron it already received the signal, it should have the signal sent through the effector, but the synapse stop this direction, stop the signal anymore. So no signal transmitted along to the motor neuron already. But for this temporal summation, meaning that they have two EPSP directly occur at the single synapse rapidly. This one is that it occur at very few period, meaning this one is at first second. After 10 seconds, E1 still contact, but the very low. But for now, when it is rapidly occur, E1 and E1 same direction, but the signal is coming through very fast in very short period, and this one we call as temporal summation. It will we can add up this membrane potential value and trigger action potential. E2 still no. Okay, for here, spatial summation is that when E1 and E2, meaning that they have two different synapses in one neuron, when they have two different synapses receive the EPSP signal at the same time, although along E1 and along E2 actually does not reach the threshold level, but they add up, we call as spatial summation, it will trigger action potential. Okay, this one is when they have the inhibitory signal, which is IPSP. So when they have the IPSP occur, even though your E1 is very strong, it will inhibit the signal, triggering the threshold level and reach the threshold level, triggering action potential. So maybe E1 alone is already strong enough, but they have I, so already stop it. So whatever you have E1 again coming along, they still cannot trigger action potential. So this is what we call as spatial summation of EPSP and IPSP. Of course, along with IPSP, of course, totally no action at all. Okay, so this is a synapse function. What is a synapse function? So you can read yourself. I think I don't need to cakap banyak-banyak. Ini awak boleh baca sendiri. Okay, next we go through the role of the neurotransmitter. So as I mentioned, you already know the two major type of neurotransmitter, right? Acetylcholine and what? Noradrenaline, right? These two are the major neurotransmitter that present use up. So in acetylcholine, normally it is for the sympathetic nervous system that you already learned in you have learned in last chapter 9. And parasympathetic is for the parasympathetic is for the acetylcholine, sympathetic is for noradrenaline, and sympathetic is for the increase the heartbeat rate, right? The acetylcholine decrease the heartbeat rate. Yeah. So first of all, acetylcholine. So what is acetylcholine? So normally acetylcholine is the chemical structure like this. This is a chemical structure, but you don't need to know. You don't need to memorize this chemical structure. So normally, when the muscle in parasympathetic and sympathetic, both of them in the middle one ganglia cell, both of them is releasing the acetylcholine, right? Acetylcholine, and the receptor name that for acetylcholine is ligand-gated ion channel and G-protein couple receptor. So couple receptor. So this is a two major type of the ion channel for the acetylcholine, and normally when they have the acetylcholine is for muscle stimulation, memory formation, and learning process. Learning process. So normally acetylcholine is found at the neuromuscular junction. Junction for ligand-gated ion channel. For the G-protein couple receptor, it is normally located in the central nervous system. Where here, this part. This part is we call as G-protein couple receptor. For the another name is ligand-gated ion channel. That one is neuromuscular junction. So what is neuromuscular junction? Later we will learn about that. Okay. For the noradrenaline or in US they say norepinephrine, noradrenaline. Okay, this one is the normally is occur at the autonomic nervous system, a bunch of the peripheral nervous system that make up for tyrosine. So this one is you can found it at the sympathetic nervous system, right? Sympathetic nervous system. Is it? Uh, so the major function normally is to increase the heartbeat rate lah. For the you already know that it is for the parasympathetic nervous system. Of course, it is normally is involved in the stress condition, highly stress condition, panic condition lah. And dangerous condition, all that. Okay. So like the in brain function, including the sleeping, anxiety, pain, mood, all this is controlled by this. For the peripheral function, including the sympathetic response, blood pressure, glycolysis, blood flow, all that is for the noradrenaline one. And there are also another neurotransmitter is dopamine. Okay, dopamine, although it's a store, maybe you are the first time heard it. This one is the neurotransmitter. Later you will learn about the cocaine, the one of the drug, the one of the famous drug in the world we call as cocaine. You will learn about the effect of the cocaine toward the addiction, how the cocaine affect the nervous system of the human. So one of the it will affect is the dopamine. It will affect the dopamine function. So dopamine is the accessory neurotransmitter in the central nervous system, a bunch that make up for the tyrosine. So normally is maintain balance in the excitation and inhibition of the neuron, important in motor function. So you will see dopamine normally is mood and also in the motivation and reward lah. So all that. Another one is this is a inhibitory neurotransmitter, meaning that when this neurotransmitter is released, it will not trigger the action potential one. We call as gamma aminobutyric GABA. But for this one, actually it's not in the syllabus lah. In the syllabus, but it stated in Campbell. But you don't need to know the mechanism. All these the mechanism you don't need to know. You just want to know that it is one type of the example for IPSP. The previous one all are EPSP. Dopamine, acetylcholine, noradrenaline, all are EPSP. Only GABA is the IPSP, the famous neurotransmitter for IPSP. Okay. Next, we go through the neuromuscular structure. Okay. So this is a new topic to you guys that you never learned in SPM. Okay. So before you go through this topic, you imagine this first. Please imagine here. Synapse normally is at here because it is a junction between neuron and neuron. For neuromuscular junction, it will only involve efferent neuron, and the muscle. Because after efferent neuron is muscle already. So it is a junction between here. It also one type of synapse, but the target is already not the neuron, the target is muscle. Okay. Muscle. So this is what we call as neuromuscular junction. Neuro, nerve, muscular, muscle, junction structure. So this is the neuromuscular junction. So you can see here this diagram. So this is a sensory neuron, efferent neuron, right? Motor neuron. And once it reach the muscle, this part they have the junction at here. This part is we call as neuromuscular junction. When we enlarge it, it will become like that. So this is a neuron, your presynaptic neuron, and here is the muscular junction already. We call as muscular junction. So at here, we still at the gap here junction here, we still say synaptic cleft, but here is already not say as postsynaptic. It is the muscle structure we call as sacrolemma. Already. So we can see here in this diagram, you can see here, this is the axonal terminal, presynaptic. They also have mitochondria, synaptic cleft, and in the sacrolemma, they have the T tubule at here. This is very important one. T tubule at here. And D is the synaptic cleft. So this is overall about the structure of the neuromuscular junction. So of course you need to know how to explain lah. Based on this diagram, you need to how to know explain lah. So it consists of a presynaptic membrane, but the postsynaptic neuron now is called as sarcolemma, plasma membrane of the muscle cell. This is a scientific name of the muscle cell. And then the presynaptic membrane will synthesize neurotransmitter acetylcholine in synaptic node with membrane closed vesicle called synaptic vesicle. So in neuromuscular junction, your neurotransmitter use is acetylcholine. You must use acetylcholine. Acetylcholine. Example, one of the example. Okay. Okay, before that, in this chapter, topic, you will learn the contraction of the muscle. We know that when we receive the impulse, what the muscle do? Your muscle do contraction, right? So how the muscle contract? This is what you will learn. So you will learn the basic structure of the muscle cell we call as sarcoma. Maybe some of your school teacher you already touch this topic in semester 1, but I think only me, because in my sem 1 video, I got touch something here, but maybe your school teacher know. But here now, you will learn about the basic structure of the muscle cell, sarcoma. Okay, what is sarcoma? Okay, this is the skeletal muscle. So in our syllabus, we only learn about the contraction of skeletal muscle. For smooth muscle and cardiac muscle, we don't learn. Not in our syllabus. We only learn about skeletal muscles, which is a majority part of the muscle in our human. So this is our muscle, right? So when you take out one of the fiber bundle of muscle fiber here, like a pipe, you will see they have many single muscle fiber at here. Right, you can see there are many single muscle fiber cell at here, and in one single muscle fiber cell, they have many myofiber fiber. So in example, in our skeletal muscle, they have many bundle of muscle fiber in one muscle cell, and in the bundle of muscle fiber, one line of muscle fiber, they have many single muscle fiber at here. Many. You can see there are maybe 10 at here. In each of the single muscle fiber, they have many myofiber. And in the myofiber, they will have the basic structure of the skeletal muscle we call as sarcoma at here. This is very important in muscle contraction because this structure will change during the muscle contraction. So this one is more scientific. Skeletal muscle, then you they have many bundle of muscle fiber. This name is the more scientific name. Okay, muscle fiber, and then in one muscle fiber they have many myofiber. This myofiber is called as sarcoma. And the muscle membrane of the cell and the muscle membrane of the muscle fiber is called as sarcolemma. Yeah. Okay, so this one is more detailed. Okay, when you go through sarcoma, okay, what makes sarcoma? All this is what? So in general, sarcoma is composed of two major components we call as thick filament and thin filament. Thick filament or myosin filament, thin filament is actin filament. So this is two major components that make up of sarcoma. And I think this thick and thin filament is one of the components that you mentioned in the cytoskeleton, right? Cytoskeleton. You still remember what is cytoskeleton in semester 1? And cytoskeleton, they have myotubule, right? Myotubule, microfilament, all that. One of the filament is thick filament and thin filament. Okay, we will go through what is thick and thin filament. Okay, so you can see here, this is a portion of the thick filament. So thick filament, normally it containing the actin binding site, ATP binding site, myosin molecule. Okay, what is actin binding site? What is ATP binding site? Okay, if you see the terms binding site, meaning that this is the place to bind actin. Meaning that here, actin binding site, this 2 is the place to bind the actin. Okay, what is actin? Actin is the component in the thin filament. So in thin filament, they have 3 major components: actin, troponin, and tropomyosin. 3 protein component: actin, troponin, and tropomyosin. So actin is this green color one, like a ball one. You can see like a ball one, green color like a ball. Troponin is this yellow color one. Okay, so what is this? This. So normally, muscle is made up of many muscle cells that are organized in bundle of muscle fiber, led by the connective tissue and collagen fiber lah. So, in thick filament, it normally consists of mainly protein myosin here. All of this is myosin one. Myosin. So thick filament is very easy. Only myosin only. They only have myosin. Just that they have the structure we call dia akan dekat sini, and 2 major structure is actin binding site and ATP binding site at here. So normally thick filament is the long rope shaped region called myosin road and myosin head here. This one is we call as myosin head. Myosin head. Head. And myosin rope adhere to the tail or of other myosin binding together with thick filament. And myosin head can bind with ATP and hydrolyze ATP. And it also contains actin binding site where it binds to actin filament to form co-spooge. Okay. So all these actin binding site and ATP binding site is very important during muscle contraction. So I just give you a simple imagine conclusion. In general situation, this myosin head actually is lay down one. Lay down one. Not it will not straight up. Lay down one. Only it will straight up when it is in excited state. And when it is straight up, ATP binding site, your ATP will bind it at here so that it straight up. And the anti-actin binding site, it will bind to the actin molecule at here. Bind to this actin thin filament. Okay, so thin filament, as I mentioned, they have 3 major component. So the majority is actin, and we have troponin and tropomyosin. So in muscle relax, not contraction, the actin is actually they have many holes at there. Dia ada banyak lubang dekat sini kan, awak boleh tengok. So it is made up of 2 helical polymer strands that are coiled around one to each other. And it consists of tropomyosin. That is regulatory protein which bound along the length of actin. So meaning that this tropomyosin is to prevent the exposure for this hole. So because why? All these holes, semua lubang-lubang ni adalah binding site for myosin here. So this dia akan macam awak boleh masa macam awak main ball dia, main hoki main hoki ada ni bola kena masuk lubang ini during muscle contraction. During muscle contraction, like this is the football, the ball, and this ball need to enter into this hole, something like that something like nak masuk lubang ini, so that the muscle contraction is completed. But for now, in normal situation, the tropomyosin is block this hole one, so that the actin, the myosin head, the actin myosin head cannot bind to it. Dia tak boleh masuk lah. Dia ada satu benda stop dia henti dia, tak boleh masuk sekarang tak boleh masuk. Hanya beberapa situasi lepas tu saya akan terangkan baru boleh masuk. Itulah maksudnya. Okay. It also consists of a troponin complex, also a regulatory protein that bound to the actin molecule. So ini dia macam screw lah. Okay, awak boleh imagine dia macam screw untuk tekak itu line tu supaya dia tak boleh bound to the myosin head lah. So yeah. So troponin complex consists of binding site of calcium ion. Okay. calcium ion. So calcium ion is very important during muscle contraction. So please important this. So this is a basic structure of sarcoma. Based on the thin and thick filament. Thick filament. This structure is very important, especially what I say, binding site for myosin, ATP binding site, actin binding site. Please remember. So I just give you a conclusion. In muscle contraction, ini dua-dua bola ni dia nak masuk lubang ini. Tapi kalau nak masuk lubang ini, awak kena buangkan line itu, tropomyosin and troponin. You need to remove this tropomyosin and troponin first. Baru then this head can bind into the actin. Ya, ini simple, sangat simple. Saya rasa saya dah cakap bahasa simple lah untuk korang, so harap boleh faham lah. Okay. And next is ini. Ini juga sangat important kerana normally structure question come out. What you need to know is the labeling. This is one sarcoma. So in general, in your school examination, maybe it will come out very more one. So maybe it will come out here, maybe come out here. Here also have another sarcoma, here also got another sarcoma. But this is the structure of sarcoma. So you can see here, the purple color one here, this is we call as thick filament. Here is green color one, is thin filament. So thick filament is myosin. Thin filament is actin. So because some of the question normally they not say thick and thin, they say actin and myosin. So you need to know what is actin, what is myosin as well. Okay. So you need to know the structure. So first of all is H zone. Okay. H zone is the region between this line here until here. It's a narrow and light area in A band, made up of only thick filament here. And then there are also I band here. I band is only have the thin element only. Only have thin element. And then they have the M line. M line is very easy. It is a middle line at here. Middle line. And then there are also the A band. A band is only have thick element. Not only thick element, it is the overlapping. You can see it have the overlapping between the thick and thin filament. And they also have Z line. Z line is only have thin element that attach together at the sarcoma end. So they have Z line, like I band only have thin filament. A band is majority of thick filament, and there are also some part of the thin filament at there. And we have also M line. M line is a middle line, and also has H zone. H zone is the biggest part one. Oh yeah, that's all about the structure. So this one is another structure of the actin. So that is why I say that during the muscle contraction, it will expose the myosin binding site like this. In the general condition, relaxed condition, your tropomyosin and troponin complex all are cover the whole of the actin one. Cover the myosin binding site one. But when they have the calcium ion bind to the troponin complex, because troponin complex containing calcium binding site, bind to the troponin complex, then it will change the structure of the change the structure. Move the tropomyosin, so that it exposes the myosin binding site of the actin, so that the myosin head can bind into it. Myosin binding site, this part is the place for myosin head to bind into it. So yeah, something like that. Okay, now we go through the mechanism. Muscular contraction, how the muscle is contract, based on the sliding filament hypothesis. Okay, so this is a structure, right? This is a neuromuscular junction. And then after that, you will do all this. Okay, first of all, when you write the essay, maybe want to write about the sliding filament hypothesis. What you need to write first. Firstly, in studying for filament hypothesis, most important thing is you need to write acetylcholine. Acetylcholine is released at the synaptic vesicle terminal. Very important. You need to use acetylcholine. Please don't write neurotransmitter. If you write neurotransmitter, no marks at all. Of course, no marks at all. Is acetylcholine released at the synaptic terminal? Okay, once it released, it will bind to the receptor protein. This receptor protein you don't need to write because different type of the skeletal muscle have different type of receptor protein. You just write general name receptor protein is okay. You don't need to write the scientific name because different type of the muscle have different type of the receptor protein. Right, bind to the receptor protein and it will trigger the action potential. Right, it will trigger the action potential, and this action potential will be propagated along the sarcolemma, down to the T tubule. This is the function of T tubule. Because T tubule is the place to transmit the nerve impulse into your sarcoma. Into sarcoma. Okay, so this tubule was transmitted along into the sarcoma. After that, calcium ion. Where is a calcium ion come from? So your calcium ion is normally stored at the sarcoplasmic reticulum. So what is sarcoplasmic reticulum? You still remember in semester 1, chapter 3, organelles, endoplasmic reticulum. We have two types of endoplasmic reticulum, right? Rough endoplasmic reticulum, smooth endoplasmic reticulum. Sarcoplasmic reticulum is one type of the smooth endoplasmic reticulum and spatial only found in the skeletal muscle. So this lah. And you already know one of the function of smooth sclerosing reticulum is function in muscle contraction and secrete calcium ion, right? But at that time, you are blurred, right? Why you say that you can function in muscle contraction? Yeah, you will learn. So this is a function. It will secrete the calcium ion. Once they have the T tubule, the nerve impulse is transmitted along the T tubule, and this T tubule normally is same location at the sarcoplasmic reticulum. So when sarcoplasmic reticulum will receive the action potential, and it will trigger the opening of the calcium ion channel. Right here, calcium ion channel will be open, and the calcium ion will diffuse down the concentration gradient into the cytoplasm by facilitated diffusion. Diffusion. Okay, once the calcium ion channel, inside the cytoplasm, this calcium ion will bind to the troponin complex here. So it bind to the troponin complex. This troponin complex, the tropomyosin and troponin complex will be rearranged to expose the myosin binding site in the actin of the actin filament. So that's all. And then after that, your myosin head will bind to the myosin binding site found in the actin, and will do the muscle contraction. So this is a summary process. Now we go through the detail. So this is a more detailed part in the neuromuscular junction. What is happening at here? The more I just enlarge the diagram. So once the neural impulse reach here, once the action potential is already reached the sarcoplasmic reticulum, it will trigger the release of the calcium ion. So if they have no calcium ion, no muscle contraction. Why? Because calcium ion need to expose the myosin binding site. If no calcium ion at the cytoplasm of the muscle cell, then no anything, no such thing, no muscle contraction. So that's all. So this is the detail one. Okay. So at the first step, when the calcium ion already come out, your myosin head is bound to the ATP first. Just now I say about the calcium ion bind to the troponin complex. That's all is about the thin filament part. It is all happening simultaneously. In examination, you can write right, until this part first calcium ion channel expose the myosin binding site, then you stop at there, and then at the same time you need to write about this. What is happening is that your myosin head here is in the low energy configuration state, is lay down one. And here for in relaxing state is lay down one. Not say lay down lah, but in scientific term is you need to like low energy configuration state. It will bind to the ATP. And it is in low energy configuration. And once it bind to the ATP, okay, it was starting to become high energy configuration state, which already hydrolyzed the ATP become ADP already. Because once it bind to the ATP, hydrolysis occur, your myosin head will hydrolyze the ATP to become ADP and phosphate. And at the same time, you already expose the myosin binding site. Your troponin and tropomyosin complex is rearranged and expose the myosin binding site in the actin, so that this myosin head was starting to bind to the myosin binding site of the actin filament. Please don't use hole or not say myosin head bind to the hole in the actin. No. I mention hole is let you us to easy to understand. Jangan cakap awak masuk lubang ini bukan lubang ini refer as myosin binding site. So this myosin head was starting to bind to the binding site, myosin binding site lah. Yeah. Okay. And then the third step is once it bind the cross bridge will be starting to form here, like down. Myosin head bind to the actin filament, forming a cross bridge with the thin filament like this. Because at the initial part, your thick filament does not bind at here. But for now, yeah. And then after that, what happened is that your myosin will release the ADP. For now, ADP still bind to the myosin head because it is required to maintain the high energy configuration. It will release. Once it release, it will trigger the power stroke. That slide the thin filament into the center of the sacroma. So dia akan macam ni. Dia akan awak dekat sini kan, dia akan push dekat sini. You can imagine that this myosin head akan menyebabkan pergerakan thin filament ke kiri. It will bring the movement of the thin filament to the left hand side. Pull the thin filament, just only using this myosin head. So for now, your myosin head will return back to the low energy configuration state. And then when the presynaptic neurons stop release the acetylcholine, your sarcoplasmic reticulum will begin pump the calcium ion back into the sarcoplasmic reticulum through the carrier protein. So this one is here. What it mentioned is here. So once the nerve impulse is already stopped, no action potential anymore, all this calcium ion will recycle back to the calcium sarcoplasmic reticulum through the active transport. They have the calcium ion pump at there. Okay, so after it move to that, move the thin filament to the center of the sacroma, then it will become back to the low energy configuration state, and this myosin head will lay down again. It will like this, lay down again, and then they will have another 80. For now, your myosin, your cross bridge is already broken. No cross bridge anymore. And your tropomyosin, at the same time, once it back to the center and it already lay down, your tropomyosin complex was starting to block the myosin binding site again. Here, this part, starting to make it like this again. Because it exposed. Once it already bind into it, already shift the actin filament toward the center of sarcolemma, then sarcolemma, once the myosin head is already released out, then the coponic complex was starting to rearrange back again and block like this, block the myosin binding site again. Okay, and then at the same time, ATP, another type of the ATP is coming into the myosin head and bind into it. Once they have a calcium ion. Once the myosin binding site is exposed. If there are no myosin binding site, then no ATP lah. Even got ATP, they still need to wait until exposure of the myosin binding site at all. If there are no myosin binding site, they just maintain like this lah. Yeah. So that's all about the mechanism. And then, so this is the diagram you need to know when this is a normal situation. Relax. So this is the H zone. So this is the H zone. And just now sorry, H zone is here. Is only have thick filament here. Thick filament, not here. This is the sarcoma. This one is the sarcoma. H zone is only have thick filament at here. And dark A band is including thin and thick filament. H zone only including thick filament only. So after muscle contraction, you need to know what happened to all these structure. What happened to jet line? What happened to M? What happened to all this? So in general, normally only I band, A band, and H zone is affected. Z line not affected one. Z line is still maintained at there. M line not affected one. Still maintained at there. Okay, what happened? You can see here. What happened to the H zone? H zone becomes smaller because during contraction, myosin head is already pull the thin filament to the center. So you can see here, in general, here is kosong line, white space. But for now, it already become back to each other. So H zone become smaller. And A band still remain the same, but I band also becomes smaller already. Because I band is the thin filament, right? Because thin filament it will already move toward the center, so your I band also becomes smaller. But A band remain they don't change. They remain constant. Z line remain constant, but only H zone and I band are changed. So this is very important. In observation maybe, or objective question or structural question, it come out what is happening during muscle contraction to the sarcoma. What is happening to H zone? What is happening to I band? All that. Okay, so that's all about the muscle contraction. Next is about the drug mechanism action. So there are two types of the drug that you need to learn is cocaine and Cuvée. So this is cocaine. Okay, so how cocaine make you addiction? Why we say cocaine is bad? Is can say that it is very dangerous drug and can cause addiction to you. Okay, so normally in normal synapse is this one is normal synapse, especially in your brain, for the neurotransmitter we call is dopamine. Okay, in normal situation, like normal, dopamine is released, bind to the receptor, and once it bind to the receptor, it will be recycled back into presynaptic neuron. Dopamine is recycle back one. Dopamine bind with a transporter green color that shuttle it back to the presynaptic terminal. Right. However, in cocaine is will block the uptake of the dopamine. Wow, how it block? Because it is the act as an inhibitor of this dopamine uptake channel. So as a result, the absorption of the dopamine is blocked at all. So what is happening? Meaning that your dopamine is accumulated at the synaptic cliff, and this dopamine they cannot be removed anymore. So that what happened is the dopamine will bind to the receptor again and again. Even though it already bind, it only bind once only. Once only. Once it bind, it need to return back. But for now, it cannot. It maintain at there. So this dopamine just their job is just bind to the receptor. Dia tengok receptor tu kosong, okay, dia pergi je. Pergi je. Lepas tu release back. Lepas tu pergi lagi. Bind and bind again. So it will bind to the receptor again and again. Never stop one unless this dopamine is reduced. So and this one will make the situation more worse because when they have another signal again, another batch of dopamine is accumulated here. So you can imagine that your dopamine is only can be increased, not decreased. Or the concentration of dopamine at the synaptic cleft only in large amount. Large amount along the time. As the time pass, your dopamine will become more and more at here at the synaptic cleft. So this will firing the pleasure pathway more and more frequently. So your central nervous system is also very smart. Why? It also know, ini tak normal lah, ini tak normal, ini tak logic. So how it will cause it, how your central nervous system your body do, it will reduce the dopamine receptor here. So in general, you have 1, 2, 3, 4, 5. Because of this accumulation, it cut out. Make the dopamine receptor become only 2. Cut down. Because it want to reduce the action potential of the postsynaptic neuron. If not, your action potential, your postsynaptic neuron is already activated, even though there are no signal in presynaptic neuron. So that's why people that consume cocaine is very hyperactive one. Very always in high very honeymoon condition one, very happy, always very happy because their postsynaptic neuron is always activated. But the body also know they want to reduce this condition, so they reduce the dopamine receptor. So what is happening? When you drink, when you stop the cocaine, so that the normal signal come out, they still release same amount of dopamine. Maybe in normal situation, it release 10 dopamine and they have 10 dopamine receptor in normal situation, like us, normal. But for now, because of you doing the consume the cocaine, now they have 100 dopamine accumulated here, and you only still have 10 receptor, and this 10 receptor maybe in 1 minute, in normal situation, 1 minute just be that once only. But for now, 1 minute it will bind to the 10 times, 100 times at all. So your body reduce the dopamine receptor. Okay, so still normal. When you consume cocaine, still normal because dopamine still at here. But once you stop the cocaine, this channel is already unblocked. When you release the 10 dopamine at the same time, still same, but binding to it only have 1. In normal situation, you should have 10 dopamine receptor, but for now, you only have 1. Only 1. So only 1 bind to eat this receptor. But if only 1 receptor, meaning that you want to trigger the action potential is difficult. Compared to, you need to know EPSP and IPSP. Just now I say because only if the EPSP only 1, unless it is very strong signal, if only 1 EPSP actually want to trigger action potential, is very difficult. Because only one receptor. Only one receptor, and then other neurotransmitter will be dopamine will be recycled back because it cannot bind with no dopamine receptor at all, and so this person will become very low mood. So what happened to it? It require more and more dopamine. So how to increase the dopamine concentration? I eat more cocaine. Because in normal situation, it only can release 10 dopamine only. In normal situation. If you want to accumulate more and more dopamine, you only can consume cocaine. And you consume cocaine again, it will reduce the receptor of dopamine again. So this is a bad recycle. You can say that it is a bad recycling. You cannot. So that's why that person is addicted with cocaine. If they don't know how cocaine, their mood they cannot control their mood. They control their mood because dopamine is controlling the mood one of the hormone, neurotransmitter, they control the mood one. Because of this, that's why the addiction can be occur. 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 So if you so you can say, but the body still can produce receptor, right? But yes, but it requires time. The time is lengthy. So it is not that when the time you wait, you still require dopamine one. You still require dopamine. How to accumulate your dopamine? You only can eat cocaine only. So this is a bad recycling. So this is a cocaine mechanism. Yeah. Okay, another one is QV. So this is another drug from the plant. It is a poisonous drug from the that be taken from the plant. So normally it is used in the hunting, especially in the old generation, very in the last century, for the person that live in the jungle, they live in the jungle, they normally use QV. So how does this affect the human? Normally kill the animal. How it kill the animal? So normally QV has the similar shape of to the acetylcholine. You still remember acetylcholine play the major role in muscle contraction. In neuromuscular junction 1. So it acts as the inhibitor of acetylcholine to bind to the acetylcholine receptor. So in the neuromuscular junction for QA, instead of binding to acetylcholine, it bind the QA, but the QA cannot trigger action potential. So this receptor not activating the sodium ion channel. So no sodium ion channel move into the sarcolemma. So no action potential in the sarcolemma. So no calcium ion. So that person, that animal cannot do muscle contraction. And you can say that all these mechanisms, myosin, tropomyosin, myosin head, myosin do not expose, no calcium ion, all that cannot, no activator, and then die. So the muscle is unable to contract in a condition we call as flaccid paralysis. So that's all about the QA mechanism. So that's all about the first main topic for this chapter. Okay, so yeah, very long, right? Okay, next we go through the second topic which is hormone. Okay, so this is the definition of hormone. So of course, all these hormone you should know lah. Because why I need to use hormone? Because in the chapter reproduction, next chapter, and also chapter homeostasis, the next two chapter, you will learn about many hormone. So why you need to know it? Because in this chapter, hormone, you will learn about for human, you will learn about the steroid hormone and non-steroid hormone mechanism. So in examination, maybe it will come out the question that related to each other one. Maybe it come out oxidative, endogen or estrogen. Estrogen is steroid or non-steroid. You don't know. If you don't know, then nobody can help you. So you need to know, especially the hormone that you always use, like adrenaline, no adrenaline, so ovary, estrogen, progesterone, FSH, LH, ADH, and then FSH, LH, and then ACTH, this one prolactin, and increase insulin and glucagon, all that hormone you already know, right? Okay, so this is a definition of the hormone. Okay, so as I mentioned, in hormone we are classified into steroid and non-steroid. Of course, you need to know, especially the hormone as I mentioned, like you will learn that prolactin is non-steroid, is peptide hormone. So other than steroid hormone, all are non-steroid lah. Peptide hormone, ACTH, non-steroid, FAH, LH, ADH, oxytocin, all these non-steroid. And then the hormone aldosterone, aldosterone is steroid hormone. No adrenaline is non-steroid. Insulin, glucagon is non-steroid. Testosterone and estrogen, progesterone are steroid. So these are the hormones that you will learn in this semester. Other than that, it's not lah. Are not in the syllabus. Okay, so what is the mechanism in the involved in the steroid hormone. Okay, you know that steroid hormone, when it's secreted from the any gland, endocrine gland, it will transported through the blood and go to the target of the organ. Okay, once it goes through the target of the organ, how does this hormone affect this organ, affect the cell? So in steroid hormone, for example, like sex hormone, progesterone, estrogen, and also the aldosterone, this hormone, steroid hormone that be through the gene activation. Okay, how does this occur? So normally when the hormone, for example, for this hormone is aldosterone, once it near the target cell, it will diffuse, pass through the plasma membrane, directly pass through the plasma membrane. Okay, what happened is that because for steroid hormone, you will still remember steroid is what type of lipid, right? Cholesterol. You will learn in semester one. Semester one is it is lipid. So it is lipid soluble, meaning that it easily to diffuse through the phospholipid bilayer one. Once it entering into the cell, it will bind to the receptor protein. It will bind to the receptor protein in the cytoplasm and to form the complex we call as hormone receptor complex. And this hormone receptor complex will enter into the nucleus of the cell. Nucleus of the cell. And this hormone receptor complex will bind to the specific section of DNA gene. Specific gene. Bind to it. And the bound protein that act as the transcription factor will stimulate the transcription of the gene into the mRNA. So it involves transcription and translation that you already learned in semester 1, chapter 2. You need to know all the things that you learned in semester 1 are relating in this semester. Because semester 1 is basic. Until from chapter 1, right? Chapter 1, you got already learned what you learn in semester 1, chapter 8, chapter 9, chapter 8, gaseous exchange, chapter 9, especially last chapter. So transcription process. After transcription, it will do translation protein to produce protein. So this is what we call as gene activation. It activates the gene to make a gene activate so that it will produce a new protein. Meaning that this gene normally is not activated if there are no hormone. When they have the hormone, then it will activate. So this is why we say as gene activation. Okay, another one, this is very simple. For examination, you don't need to write the transcription mechanism and translation mechanism. If the mention want you to write explain the mechanism action of the steroid hormone, you don't need to write the mechanism for transcription and translation in very detail. Don't need. You just write got transcription and translation. That's all enough. Don't need to write detailed one because this chapter is not examine your transcription mechanism theory and translation mechanism theory. No, they want to examine these three parts. The location, especially in the cytoplasm and in the nucleus, you need to mention clearly. If you not mention the location, no marks at all. About next is non-steroid hormone. So non-steroid hormone is more complicated compared to steroid. Because non-steroid hormone normally is derived from protein, so they cannot easily pass through the plasma membrane one. Not like the steroid hormone, because it is lipid insoluble. Because we know that phospholipid bilayer is made up of lipid, right? So because it is lipid insoluble, of course it cannot easily pass through into it. So how does this happen? So normally in non-steroid hormone, you will use this term as the signal transduction pathway. Meaning that this is a general theory, meaning that this is a hormone, your hormone, it binds to the receptor. So this is a signal reception. After that, it will go through along the series of the chemical reaction we call as signal transduction to do a cellular response. So sometimes occur in a single step, but more often require a sequence of a change in a series of even molecule. So meaning that this hormone bind to this receptor, after that you need to change this molecule again, you should change this molecule, finally through the cellular pathway. So it's more complicated. We call a signal transduction pathway. These are general terms. In examination, don't not say that non-steroid mechanism is this no, this is the scientific name of the mechanism for non-steroid hormone is second messenger CAMP here second messenger CAMP. Okay, how does it occur? Okay, first of all, normally you will the hormone, the non-steroid hormone will act as the first messenger first. This first messenger first bind to the GPCR, G protein couple receptor on the plasma membrane of the cell. After that, this GPCR is activated. It activated GPCR. Okay, what happened to this GPCR? GPCR will bind to the GTP, bind to the G protein, which is then bound by GTP, activating the G protein. Okay, this GPCR will bind to the G protein here. This G protein is inactivated. So bind to the G protein, at the same time, your GTP will bind to it to activate the G protein. So, but to able to move the cytoplasmic membrane of a membrane, G protein function as a molecular switch that is either on or off, depending whether GTP or GTP is attached to it. So when GTP is bound to the G protein is inactivated. So when this GPCR is activated, it will bound to the G protein, and GTP bind to it as well, so that it become activated. So G protein is activated. So this G protein, what is do? This G protein will activate endogenous cyclase become activated. I'll repeat again. G protein will bind to the endogenous cyclase. GTP is hydrolyzed, activating the endogenous cyclase. And this endogenous cyclase will convert the ATP to CAMP. So this CAMP is act as the second messenger to activate another protein for cellular response. So this is very complicated. So first of all, the hormone, the non-steroid hormone bind to GPCR first. GPCR bind to G protein, activate the G protein, and this G protein activate the endogenous cyclase, and this endogenous cyclase but convert the ATP to become CAMP, and this CAMP act as the second messenger to activate the other type of protein, and this protein will activate the cellular response. So one of the example that is mentioned in Campbell is adrenaline. So you can see here, G protein, inactivate G protein. Once it bind by the GPCR, it become activated. And this activator bind to the inactivate endogenous cyclase become activated. And this activated endogenous cyclase convert ATP into the CAMP. So this CAMP act as the messenger. So this CAMP will activate the inactivate protein kinase A become activate protein kinase A. The most important term is inactivate and activate. You need to say cyclic AMP act as second as messenger, activate inactivate protein kinase A becoming activated protein kinase A. After that, protein kinase A activate inactivate phosphorylase kinase to become activated phosphorylase kinase. And then activate phosphorylase kinase activated inactivate glycogen phosphorylase becoming activated glycogen phosphorylase. And then this Inactivated glycogen phosphorylase, activated glycogen phosphorylase will contribute the cellular response, which hydrolyze glycogen to become glucose 1-phosphate. This is all the entire mechanism for using example as adrenaline. The most important thing is here. You need to mention inactivate, activate, becoming activated. This active term is very, 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 very important in mentioning non-steroid hormone. And you just mention, don't memorize only adrenaline is enough lah, because Campbell only use adrenaline as example. So you need to, you don't think not come out, 1 year come out already, in past year come out, directly using adrenaline and directly want to explain how this adrenaline trigger the cellular response. You need to all mention all this. Normally, you only mention about protein kinase A. Most of your students memorize only until protein kinase A, and then after that cellular response. Meaning that in adrenaline, they just say, activated protein kinase A, then directly go to cellular response, convert glycogen, and don't use convert. Please don't use convert. Don't use convert. Not say, cyclic AMP convert protein kinase A becoming protein kinase A. What are you talking about? So please, you don't use convert. You need to use activate. Activate verb, and then activated is the terms. And inactivated is the terms. For example, cyclic AMP act as second messenger. Will activate inactivated protein kinase A becoming activated protein kinase A. You need to mention all the terms. Inactivated, activating, activated. All this you need to mention one. You cannot miss out any terms. If you miss out any one of these terms, no marks at all give you. I know it's very complicated, but I cannot tell you because this is biology, this English, So I really try my best, so hope you know. Okay. Next, we go through the hormone part, plant hormone. So that's all about human hormone. Human hormone, just two things only. Mechanism of steroid hormone and mechanism of non-steroid hormone. These two things only. Next, we will proceed to the plant hormone. So in our syllabus, there will have to 6 types, if not mistaken, is 5 types of the plant hormone that you need to learn. First one is auxin. Second one is gibberellin. Third one cytokinin. Fourth one is abscisic acid. The fifth one is ethylene. And then got another one, the new syllabus one. In the new syllabus, they add on the another hormone is recalled as barsinosteroid. The barsinosteroid is in the new syllabus one. In the old syllabus, only have 5 types of the hormone, but in new syllabus, they add on the barsinosteroid. So first of all, auxin. So auxin, I think in SPA you already learned, right? What is auxin? What is auxin? So the general function of the auxin, I think everyone is already know. So this is a basic explanation about the auxin. So what the synthetic auxin, naming synthetic auxin means that it is the human made auxin, is recalled as IAA. So if you see the terms IAA, the full name is indoacetic acid, is a type of the auxin. Actually, they have many names one. IAA is one of the famous one. And normally it is secreted in the shoot and root, apical, meristem, and young leaf. So for the plant hormone, of course, you need to know the location where does this hormone is secreted, their function, and also their function. These two is the major component. And then transporter, blah 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 blah, and then not important. This one phototropism for auxin is very special one. The growth of the plant toward the light or away the light is called the phototropism. So normally for auxin in the shoot, the cells on the darker side will elongate faster than the brighter side. So I think this one everyone know, right? And so they will have the positive phototropism occur in auxin is where the high auxin concentration is accumulated at the darker side of the stem, stimulate the growth and bend toward the sunlight. So this Still no light, phototropism, meaning that when during the sunlight, when you have the sunlight, your auxin normally is moved toward the away from the sunlight. What is at here, right? So the rapid growth is at here. So that's why I say that positive accumulate at the darker side of the stem, stimulate the growth and bend toward the sunlight. So this what lah. And another one is geotropism, right? Geotropism is they accumulate at the accumulate at the lower side, higher oxygen concentration cause more rapid growth on the lower side. The tips go curve upward. But for the root one, oxygen is accumulated at the lower side, but slow growth. So in general, from this experiment, you can know that oxygen provide growth for shoot, inhibit growth for root. Right. Right. So I think this experiment or everyone is already know, right? Charles Darwin experiment for the oxygen one. So it need to want to determine know how the plant bend toward the light, because we know that in your primary school you already know plant is bent toward the light one. When the light is at here, then your plant is bent like this. When your light is at here, then your plant is bent toward to the left hand side. Why? The scientists need to know why at that time. So that one of the scientists called Charles Darwin, they do this kind of experiment. So the first one is control. They do control. They do not have any adjustment for this plant. For the second one, they remove the tips, because we know that oxygen is secreted from the meristematic, normally is located in the tips. So once you remove the tips, no any response at all, because no oxygen is secreted. So the plant not growth. They just maintain at there. No growth at all. But for control, of course, you see the plant growth, bend toward the sunlight lah. Yeah. And then after that, they use the they use the black color cap at there. Means not untransparent, black color, totally black color. And then they see that the plant growth, but they growth straight forward. They do not bend one. And then for this one, they use the transparent cap. They found that it go bend to the sunlight. For the fifth diagram, they cap cover on the stem of the plant instead of the cap instead of the tips but still the growth toward the sunlight. So this experiment conclude that the oxygen is secreted at the tips, meristematic at the tips of the shoot, and also the oxygen is have this kind of negative geotropism of the shoot on the shoot, and positive phototropism on the shoot So this is the experiment, and another experiment by Jensen. Okay, this experiment is to determine the location of the oxygen. So for this experiment, when they use the mica barrier inserted in unilaterally, when this mica barrier is inserted at the left hand side, or imagine the sunlight is at here. When it inserted at the left hand side, because we know that oxygen is bind toward at here, bind to the shaded region. So because if you put some of the barrier at here, so oxygen cannot accumulate at here, so no bending at all. But when you do the barrier at here, since oxygen still can accumulate at here, so the binding can happen. And also this one is another experiment. They cut off the teeth, but then they cut off the teeth, but then they put the gelatin barrier at here and put back the tips at the upper side. They can still can bend to it. So this mean that oxygen still accumulate at here, and oxygen still can diffuse into the through the gelatin barrier. For the veins, it need to determine by cutting the tips of the plant. But however, the hormone from the tips is stored in the agar block. So the agar block will place on the gut part here. So the collotype will bend toward the light. Auxin is moved down the shaded region for the spot of gold. So we can see here when they cut down the tips, and they put the hormone in the jelly, and this jelly is put back into the tips, and they found that it still can grow. So that this hormone is still is a auxin still can be function even though the tips is removed. For of course, if you're not using the jelly that do not have the auxin, of course they do not go. Okay, then another experiment by Bush. Bush one is they also use similar experiment like this, but they just using the different parameters. So for this one is the unidirection light, meaning that the light is occur at here from the left hand side, but the auxin is at the right hand side. Like this. And the same amount of total protein is produced by the soup tip go with the direction light, even when a barrier divide the soup tip. And the soup tip is grown in the dark, all three blocks of agar cause the same amount of curvature into the sun. So you can see that even though they use the three different agar plate, they still have the same the curvature is still the same. Actually, this experiment is not very important. It's not very important. Yeah, because I think this experiment you already learned in SPM. In STPM, normally they will not come out. Want you to do the experiment. Yeah. Okay. This one is very important. Low of the auxin. Okay. So how the auxin meet key function of the auxin. So in plant, so normally the function of the auxin is primary growth, cell elongation. Very important, cell elongation, meaning it make the plant cell become bigger. Okay, so how the auxin trigger the plant cell elongation? So normally, firstly, auxin will bind to the receptor of the basal membrane according to the acid growth hypothesis. This one, acid growth hypothesis. When auxin firstly will bind to the cell membrane of the plant, and then auxin will increase the proton pump activity from the cytoplasm, meaning that it will increase the hydrogen ion inside the plant, inside the cytoplasm. So when the hydrogen ion is accumulated in the cytoplasm, of course you will reduce the pH of the cell wall, is make it more acid. So this reduced pH will activate the expansion protein, which is red color here, separate the microfiber from the polysaccharide. So this means that this protein, if there are no auxin, is inactivate. When auxin present, it will activate it. So what this protein do? This protein will separate the microfiber here, all these brown color, for the polysaccharide. Meaning that the polysaccharide will be removed from the microfiber. You can see here. See here. It already removed. And these polysaccharide are cleaved by cell wall loosening enzyme. So the cell wall loosening enzyme is a purple color one. Purple color meaning that it will cut the polysaccharide. So normally the plant polysaccharide is the cellulose. And this process will loosen up the microfiber, making the cell wall more flexible. Water and increased turgor pressure will cause the cell to be elongated. At the same time, more microfiber are formed. So actually, this one is to make the cell like the cellular microfiber become loosened, become more loose, so that it is not tightly like this, so that it can expand the cell elongation more easily. So based on the acid growth hypothesis, by reducing the pH of the cytoplasm in the cell. Okay, so this is a major role. And then another role is to enhance apical dominance Okay, what is apical dominance? So apical dominance actually is the inhibition of the growth of axillary bud, or another term is lateral bud. Some of the book use lateral bud. Lateral bud. For me, I use axillary bud. Axillary bud by the apical bud of a plant shoot. Okay, this is a apical bud. So apical bud is a major part of the auxin to synthesize one. So normally your auxin will move downward for the apical bud, produce the another hormone we call as strigoglucan that replace the growth of the axillary bud. So that is why we say as hence apical dominance. So hence apical dominance. So the axillary bud is inhibited, no growth at here. Okay, this diagram is with apical bud removed, meaning that the axillary bud is already grown. Maksudnya axillary bud yang macam cabang untuk cabang-cabang lain-lain untuk plant tersebut lah. Kalau ada toksi, plant tu lebih cantik. Kalau tak ada auxin, plant tu macam tak berapa cantik lah. Dia ada sini punya tak cantik. Ini cantik. Easy to do. This is we call as axillary apical bud. Apical bud is at the at the tips part, and then you can see there are two like the leaf shape one, the light green color one. These two is we call as lateral bud or axillary bud. So when they have the auxin, they inhibit the growth of the axillary bud. So that's why we call as hence apical dominance. So if the apical bud is cut, apical bud here is cut, the axillary bud or lateral bud will go and take over as the new apical bud. It will go again, remaining bud to receive more sugar, auxin. So no auxin. So this nucleus will decline the topmost axillary bud in particular to go and take over as the new apical bud. And then after that, they will new apical bud will be formed at here. Yeah. So this is what we call as a role of the auxin. So of course, auxin also have other role. Also can alter gene expression, causing cell elongation, short life transcription factor, activating other gene, and promote food growth, induce parthenocopy, which is the food development without fertilization, inhibit leaf abscission, establishing phyllotaxy, arrangement leaf of the team, and promote cell division in leading to the secondary growth. And then this is the application of the auxin. So they are for me, I give you 4 application. So one of the application is herbicide. They use 2,4-D dichlorphenol acetic acid that can kill the herbicide one. Highly effective against the dicotyledons plants, and also root development and propagation. IAA, they use IAA stimulate the root development and stem cutting and used for the asexual propagation. And preventing the premature foot drop, you use NAA. Then prevent the formation of the abscission layer, the tissue that cause the food to detach from the stem, improve the both yield and food quality. Meaning that even though the food is already mature, but the food is not dropping And then also promoting food set and enlargement make the food more bigger, more tasty, more colorful, develop the parthenocopy without using the fertilization process. Auxin can do it. Okay, next hormone is cytokinin. Okay, so cytokinin actually is I can say is the siblings of the auxin. They normally are working together so work together. Okay, so normally cytokinin is secreted from the roots. Normally is roots and transported to the organ by the xylem vessel. And normally this is the general plant role regulate cell division. Okay, in shoot and root. So this is different from auxin. Auxin is for cell elongation. This is cell division, meaning that it can divide the cell make the cell more quantity lah, and modify apical dominance, promote the lateral bud growth, promote movement of nutrient into the zinc tissue, stimulate seed germination, delay leaf sensors. Stimulate seed germination, I am not recommending you use cytokinin lah. I am recommending you use gibberellin. Okay. Okay, rules of the cytokinin. Okay, so actually it is act as the control of cell division and cell differentiation one. So normally it is acting with along with auxin one. So you can see this diagram. When you see here, when auxin is high in concentration, it will cause shooting, opposed to development of the axillary bud, favor to the rooting. For the cytokinin, opposed to the root development, favor to the development of axillary bud is for shooting. Okay, so in general context, if auxin concentration higher than cytokinin, it favor the rooting. If the cytokinin higher than auxin, it favor shooting. If auxin equal to cytokinin, it will form catalyst. Okay, what is catalyst? Okay, catalyst, we will learn about it reproduction topic. It is the one of the structure that normally is formed in the plant tissue culture. This one is you will learn in next chapter. I will explain more about detail in next chapter reproduction. And also this is in the new syllabus at all, because in old syllabus does not have plant tissue culture one. But new syllabus they already include in basic plant tissue culture. So yeah, I will explain more about in next chapter lah. Next chapter. Okay, so normally in cytokinin, normally it promote the cell division and cell differentiation process relatively in unspecialized cell into mature, more specialized cells. And normally, cytokine is related with the auxin to the formation of the plant organ. Okay, so the differences between cytokinin and auxin is auxin make the cell more bigger, more larger, but cytokinin make the cell becoming more mature. It increase the number of the cell, so it make the cell more mature. So these are the differences between cytokinin and auxin. And another difference is the apical dominance. So for auxin, it enhance apical dominance, but for cytokinins, it inhibit apical dominance. It promote the growth of the axillary bud. It enhance growth of the axillary bud or lateral bud lah. Lateral bud. So there's that I mentioned before lah. When cytokinin is highly concentration at here, even though you not cut the apical bud at here, if the cytokinin is very high from the root, allowing a limited amount of the axillary bud to grow. Although auxin is here, but it still can grow it, but very little. Therefore, the axillary bud farthest from the apex are increasingly elongated. Of course, there are another effect of the cytokinin is anti-aging effect. When you spray the cytokinin into the leaves, it may maintain the leaf in green color one, delay the leaf sensors. So anti-aging effect like in human you can say like the antioxidant properties make your skin more collagen, make your skin more like youthful people, something like that cytokinin is for make the plant more youthful not make the plant leaf is always in green color one. So this is anti-aging effect and then this is application of the cytokinin, so it can delay the leaf sensors. So like this anti-aging effect. Anti-aging effect, it can delay the breakdown of the chlorophyll and protein in the leaf, so that it can extending the leaf lifespan and also make the plant can tolerate in drought condition because it influence the root development, improve water intake, because I already said cytokinin is secreted in root and also play the major role in cell division and differentiation for the plant. So when in the drought condition, cytokinin will make the plant is more tolerant to the drought, maybe make the plant roots more longer so that it can easily uptake the water from the underground lah. And also micropropagation and tissue culture. So this one is more focused on the tissue culture lah. Culture lah, which I mentioned before is this lah. When you using the different ratio of auxin and cytokinin, you can control the growth of the plant. Either you want the shooting one or rooting one, or even you want to form the catless. Yeah. So this allow for the rapid, large scale and disease-free propagation of the desired plant varieties lah. Okay, third hormone is gibberellin. Okay, so this hormone is majority that you will learn is seed germination. Yeah, majority function that you use is seed germination, because in next chapter one topic is about seed germination. Gibberellin is a hormone that play the role in seed germination. So the mechanism about seed germination next chapter I will explaining. And then this normally is secreted is from the meristematic of apical buds and roots. Apical buds and roots, young leaf, and also developing seeds lah. Okay, and beside apart from the seed germination, gibberellin also play the role in stem elongation. So this is the two diagram with one is got using gibberellin, one got using gibberellin, one does not use gibberellin. This is the difference. Gibberellin are the best known to stimulate the stem and go for the enhancing cell elongation one. So they can activate the enzyme, thus gibberellin can act in concert with auxin to promote stem elongation. So like auxin because auxin play the role in cell elongation, right? Gibberellin also can. Of course, the next one is seed germination. So seed germination I will explain in next chapter. So for this time I don't explain more. Okay, and okay, so this is the application of the gibberellin. So first of all is breaking seed dormancy and enhancing seed germination because you know that gibberellin can play the role in seed germination. So for normally like in extreme condition, like the very cold environment or in the light certification environment, gibberellin can can initiate the seed germination at that kind of environment. And delaying senses, like the gibberellin, it also can delay the plant senses, but normally this one is for the delaying the ripening of the fruits and maintain the fruitfulness. Of course, to improve the stem elongation and plant height. Okay, the fourth hormone is abscisic acid. So this hormone is the inhibitory hormone. Among the three hormones that mentioned just now, all the three hormones is stimulate growth one. But this hormone is inhibit the growth one. So abscisic acid inhibit growth. So almost all plant cells have the ability to synthesize abscisic acid. So you can say that almost all part of the plant can synthesize abscisic acid. And normally the major function is to inhibit growth. And as I mentioned in first chapter, can say a change in the mechanism of the closing of stomata during the drought condition, you got see the abscisic acid, right? So this is the function, one of the function. So actually you can use that kind of mechanism as the function if you want to write it. Right? And promote seed dormancy, meaning that seed dormancy is the antonym name of the seed germination, not germinating the seed, inhibiting early germination. Promote leaf sensors, promote decision tolerance. But the cytokinin delay leaf sensors, right? But these hormone promote leaf sensors. Okay, so first of all, the first one is seed dormancy. So dormancy is a definition as an evolutionary mechanism that synchronizes germination with optimal abiotic condition. So in very extreme condition, abscisic acid will enhance the seed dormancy so that the plant will not germinate. The seed is not germinating. So the high level of ABA will inhibit the germination and induce the production of protein that help the seed withstand the extreme dehydration condition. condition. Also the ratio of ABA to gibberellin can determine whether the stress seed remain dormant or germinate lah. Yeah. And also, of course, drug tolerance. It can induce drug tolerance. So this one is almost similar what I say in first chapter lah. How the abscisic acid can make the plant survive in drug tolerant condition. So normally is the closing of stomata. It will induce the closing stomata, reduce the water loss, reduce the gut cell gurgle, so leading to the closure of the stomata, so that the water cannot easily transpirate lost from the plant by the transpiration process. Okay, this is the application of ABA. So normally it's enhancing drug and abiotic stress tolerance. So it can make the plant survive at the drug condition and abiotic stress. And also cold tolerance. Cold tolerance, even at the very low temperature environment, ABA still can make the plant is survive at there, improving food quality and ripening. Okay. Next, the fifth hormone is ethylene or ethene. Another name is ethene. I would recommend you use ethylene lah. Okay, so this actually is gas hormone. Compared to the few previous hormone, normally they can be liquid hormone, but this one is gas hormone. So normally it is produced in foods lah. Normally produced in foods. Produce high concentration in during leaf sensors, leaf absorption, and ripening of the foods. Of a food. So play the major role. One of the key major role is food ripening. But in STPM, of course, you need to write some more. Not only food ripening. Of course, you need to write. And also leaf absorption, stem elongation, and hence the rate of sensors. So actually, leaf absorption and leaf sensors actually is the aging effect of the plant. So if too much of ethylene actually is you make the plant aging more faster promote root and root hair formation, promote flowering in the pineapple family. So of course the first hormone function is the food ripening So I think I don't need to mention more about these. So normally we'll break down the cell wall component of the food and then convert the starch acid to make the sugar, make the food is more sweet. So that you eat the food is sweet, right? And they also produce the color which is more colorful, and then ethylene will trigger ripening, and ripening will trigger more ethylene production. So this will result in a huge burst in ethylene production. Okay, another one is triple response to mechanical stress. Okay, so what is this? So normally this hormone can instigate a growth maneuver known as the triple response that enable the shoot to avoid the obstacle. So you can see here, when you do not have any concentration of ethylene, your plant is grow like this. But when you apply ethylene in highly concentration, you can see why the plant look like bend toward and bend, look like very shorter, right? Because they have three main response. The first response is it can slow down the stem elongation. Slowing the stem elongation. Second one is enhance the stem thickening, make the stem is more thicker. Third one is horizontal stem growth. So it is go horizontal instead of vertical. This response is greater with increased ethylene concentration. So this is one of the effect of the ethylene. If you apply too high concentration of ethylene, it will produce this kind of effect. Of course, in ethylene, it operates with aging effect sensors. So meaning that it will program death of certain plant of entire plant. If your plant have too many of ethylene, it will enhance the aging effect and your plant is easy to die And then another one is leaf absorption, meaning that your leaf is easily to be fall down. There are the difference between these two. Leaf sensors is your plant, your leaf already is dying, but maybe it's still on the tree. It does not fall down, but it already wish to fall down already lah. But leaf excision is that the leaf is already loose out from the tree. Loose of the leaf from the deciduous tree to prevent desiccation during seasonal period when the availability of water to the root is severely limited. So normally in autumn country lah, that for seasonal country, during autumn period, you can see that all the leaf are fall down because they need to survive in the winter condition, because in winter condition the plant need to get the water is very difficult, because all the plant your underground water maybe is already becoming ice already, or the surrounding land is already becoming the ice. And then this is application of the ethylene, so it can act as the control of the food ripening. So this is one of the application there where they use the gene editing lah. They edit the gene of the tomato. So this is the wild type of the tomato. They're containing the ethylene gene, and then they transcript and translate the ethylene function to produce the ethylene enzyme, ethylene, and make the tomato ripen lah. But this type of the wild type of the normal situation will make the tomato is easily to be rosak akan menyebabkan tomato ni senang untuk rosak, tak mau. So they need to delay it. How they delay it? They use the gene technology lah. So they use the gene technology. So after transcription plant, transgenic plant, both the sense and antisense sequence for the interim biosynthesis are transcribed. So they will have add on the antisense gene at here. So once in the transcription, sense and antisense gene and mRNA, they are complementary to each other. So they will bind to each other. They will bind to each other, form the two double-stranded mRNA lah. That cannot be translated. So your tomato is still not ripened at all. Cannot ripen at all. Once you transported this tomato into the market, and then you apply you spread the ethylene gas, then it would be ripened at that time. So you delay the food ripening lah. Thus, wild type tomato may already be rotten and damaged by the time they reach the store. Transgenic tomato can stay fresh longer lah. Okay, next is pre-harvest management. So normally this one is to refer to the entire period and the set of the activities that take place before the crop is actually picked or removed from the field. So normally this one is the agriculture management we call as pre-harvest lah, because we want to ethylene is able to remove the excess more food, so the remaining one grow larger and higher equality. So normally this one is at the agriculture, because in one tree actually they have many foods, but some of the food actually cannot go very nice one. So when you use ethylene, the very small food you can remove at very small foods. The size of the food is very small, can be removed to allow only the larger and higher quantity food can be grow lah. And also can weaken the connection between the stem and branch. As I mentioned, is the axis zone, so that mechanical harvested or unharvested fruit can be easily shake nuts or cherries loose without damaging the tree. Meaning that you just shake a tree easily, then the food, all the unwanted food already fall down already. And also trigger premature essential program cell death when synthesis is respond to mechanical wounding and pathogenic infection. Okay, and then this is the last hormone, which is called as bassinosteroid. And also this is a new syllabus add-on hormone. In previously they not add on. So normally this hormone actually is similar to the structure of the steroid hormone in animal, which is cholesterol and testosterone. You can see the structure is almost similar. So normally it's can be secreted in the all plant tissue all plant tissue normally such as the pollen, immature seed, shoots, and leaf lah. Something like that. And this is the role of this hormone. So normally they are promote cell expansion and cell division, and induce cell elongation and cell division in stem segment and seeding at low concentration, and promote good growth at low concentration, inhibit root growth at high concentration. Low concentration promote good growth, but in high concentration it inhibit good growth. Promote xylem differentiation, inhibit phloem differentiation. Difference. Xylem is promote, but phloem it inhibit, and also can promote seed germination and pollen tip elongation. Elongation and something like that. Okay. So this is a final hormone about this. Okay, this is the last topic of this chapter, which is the cytochrome-like response of the plants. Okay, so this is based one of the big application that you need to learn is the flowering lah. Flower. There are many factors that can affect the plant flower. One of the factor is cytochrome. So what is cytochrome? Cytochrome is the pigments that absorb in the plants that absorb mostly either red light or far red light, regulate many plant response to light, including seed germination and shade avoidance, as well as the flowering lah. Okay, so what is red light and far red light? Okay, you can imagine that in plant we have the pigment, like the chlorophyll pigment we know that chlorophyll pigment absorb the light at red light and also blue light, right? We have two type of photosystem, P680 and P700, right? So in similar, cytochrome is also like this, but we don't need to know the location of the cytochrome. You just want to know the there are two types of the pigment found in the plants, which is PR and PFR. Cytochrome PR and cytochrome PFR. So what are these? So PR, already mentioned, meaning that this is the cytochrome absorb the red light only. Okay, what is red light and far red light? Okay, red light normally is they have the sunlight. So meaning that PR absorb red light normally happen during the sunlight day, during the day. For the far red light is during the night. So imagine red light equivalent to day, far red light equivalent to night. Okay, so PR absorb red light. So we know PR absorb red light. So when it absorb red light? Only during the day, only the light. When you have the sunlight during the day, then PR is absorbed. Okay, so PR, what is absorbed? Absorb red light to what? So it absorb red light, convert red light at wavelength 660 nanometer, convert the red light into the convert the cytochrome PR into the PFR. Okay, meaning that in the sunny day, PR, we have PR. The plants have cytochrome pigment PR. Okay, this PR will absorb the red light. Okay, it absorb the red light, and itself will convert become PFR. So in simple terms, during the day, sunny day, concentration of PFR is higher than PR because majority of PR already convert into PFR. Okay, similarity for PFR. PFR is absorb far red light. So far red light is at absorption of far red light is at night only the night. So at the wavelength about 730 nanometer lah. So this one is a scientific term you need to know. Scientific lah, more scientific value. Okay, so when it absorb far red light, it will become red light. So in night, during the night, PR is higher than PFR because majority of PR already convert to PR. So this is the relationship between PR and PFR. And in general, red light will increase the percentage of the seed germination, inhibition of vertical growth, stimulating the bunching, setting the internal core, control of flowering. So in our syllabus, we were more focusing on control of flowering. Seed germination is not our control side, but you just know in general, PFR, EFR, if the PFR pigment is high, it can induce the seed germination process. That one is this one is the flowering plant, photoperiodism. Okay, so photoperiodism is the physiological response of plants to specific darkness or daylight lengths. Okay, so what are these different? This is the cytochrome. It's just a pigment that found in all of the plant. They just either PR or PFR pigment. They already found that. This one is specific for the plant, is the situation for the plant respond to specific darkness or daylight lengths. Meaning that we have two types of the plant for flowering, long day plant and short day plant. Long day plant meaning that when the sunny day, the sunny day is higher than the night period, the sunny day period is higher than night period, this plant will flower. But for short day plant, when the sunny light period shorter than critical dark period, critical line, it will flower. Only it will flower. Meaning that long day plant, when the sunny period higher than longer than critical line, this is a critical dark period or critical line, then it flower. It's shorter than it, then no flower. For short day plant, when the sunny daylight shorter than critical dark period, only it will color. So the below is the example of the long day plant and short day plant. Tomato, rice, and dandelion are the long day plant. Soybean, pocentias, and chrysanthemums are the example for the short day plant. Apart from that, there are also other type of the long day plant and short day plant for the flowering. You can Google it, but these are the three example that I can found in Canberra. So majority maybe MPM will came out question based on these three example. Because in got 1 year, the MPM does not using the terms long day and short day plant, they directly using the plant name. Chrysanthemums and another is dandelion, you're not mistaken. Yeah, they just use dandelion and chrysanthemums, and then you need to write the mechanism. So if you don't know it, although you write your mechanism is correct, long day plant and short day plant are correct, but because you already misunderstanding the dandelion, maybe dandelion is long day, you say dandelion is short day, and chrysanthemums is short day, you say it is long day. Then although your mechanism are correct, but entirely question are wrong, because the first sentence you say the statement is already wrong. In biology, it's like that. If your first statement is wrong, then entirely question, although the mechanism is correct, are totally wrong. The very strict one. Biology essay question is very strict one. So example is very important. You still need to know, including the hormone part one. Maybe you will not use the steroid hormone or non-steroid hormone, you just use progesterone. Then you need to know progesterone is steroid or non-steroid. Okay, so this is the mechanism of the far red plant or short day plant. So we see it one by one. Okay, one by one. One by one. Before that, I just want to say we want to write the mechanism about the long day plant. How long day plant flower? We know that of course sunny day plant is need to longer than late critical night length. So how when it longer, how the plant flower? So they will have a plant hormone is called as florinogen. Florigen hormone. So it will based on this PR and PFR. Okay, so in long day plant, we need to know in long day plant, you only can flower when daylight period is longer than night period. So meaning that sunny day period is mid to very long. Okay, when sunny day is very long, what happened to PR and PFR? PR is converted to PFR, meaning that the concentration of PFR pigment is higher. PR is lower. So high level of PFR in long-day plant will stimulate the flow region, the inactivated flow region become activated. So this flow region hormone become flowering hormone to induce the long-day plant for flowering. So similarly for short-day plant. Short-day plant only flower then night period longer than sunny day period. So meaning that night period is more longer. So what is happening in night period? PFR is converted to PR. So PR is higher than PFR. So a higher concentration of PR stimulate the inactivated flow region hormone become activated flow region hormone. So this activated flow region hormone will act as the flowering hormone to induce the flowering of the short day plant. Okay, so this is a mechanism how the long day and short day plant flower based on PR and PFR. Okay, normally in exam, they will come out question like this. Okay, we will look it one by one. First of all, for this, so this is short day, this is long day. But in examination, they will not come out like this. They will just come out like this, and then it will just all say you this one flower, this one not flower, this one flower, this one not flower. So this is short day plant, long day plant, or long day plant. Like they will ignore, they will blur up this. So but for the lesson purpose, so I let you know that this is short day plant, this is long day plant. Okay, you see this. When this one is the sunny day, the daylight period is longer than critical night length. So meaning that this is sunny. Sorry. For this situation, first situation, you can see that the first the sunny day period is longer than the critical night length, meaning that for short day plant, no cannot flower, it cannot flower. Long day plant, yes, it can flower. For here, you can see the sunny the day period is shorter than critical night length. So meaning that this one is short day plant will flower and long day plant cannot flower. Okay, but here you can see this is the situation same as the number 2 one. The sunny day period, light period, shorter than critical length. Okay, in theory, short day plant should be flower, but in the dark period, they flash the red light. Suddenly use the flash of the red light at here. Okay, when you have the red light, meaning that at this period, PR is converted into PFR. PFR will be more higher. PR is lower. Okay, when PFR is higher, is higher, what happened to it? If flash of the red light, meaning that PR is converted to PFR. So when PFR is higher, increase in amount, what plant is flower? Should be long day plant, right? Because it increase the PFR concentration compared to this. Compared to this. So that's why that short day plant cannot flower anymore. Because it already give a flash of the red light, meaning that this is the period that have the sunny period, daylight period at here. So long day plant flower. Okay, here you can see it's similar to previous situation, but you flash another far red light at here. So this far red light already cancel the red flash effect. So it's similar like this. It have a PFR increase, but then you flash the far red light again, so PFR decrease again. So long day plant cannot flower, short day plant flower again. So you can see here, even though it is same, and then you flash again the red light, so the long day plant flower again, and then you flash again far red light, short day flower again. So it can cancel the effect one. If the situation is the same, when there are another far red light come out, it will cancel. Similarly, if the situation is far red light first, and then far red light flash of the far red light, meaning that short day plant is flower, long day plant is close. So normally, this is the situation that you need to know. So for long day plant, normally they will more longer eat. More longer. For example, for this situation, you imagine that the sunny period is longer than critical period. It should be long day plant flower and short day plant not flower, right? But at here, I flash the far red light at here. So it will opposite lah. It will become that long day plant cannot flower, short day plant become flower. Yeah, something like that. So this is the critical thinking question type, and normally will come out in objective lah. And even though it will come out in subjective, but essay question very rare lah. When essay question normally come out like this mechanism lah. But in objective or subjective question come out normally is critical thinking. You need to know whether this plant can flower or not. Normally the leaf leaves, and then it will ask you, can this plant flower or cannot? For subjective question, of course, not only ask you, but it will ask you explain why. So that's all for this chapter 10, control and regulation. And also hopefully this video, free class lesson video can provide the knowledge to you guys, understand easily what you will learn for this chapter lah. And good luck for your biology examination, and also hopefully you can master well for this chapter. And if you have any question, you can comment to the chat box, or you can join the group to ask any question that you want. So thanks for you guys watching, and thanks for your guys support, and thanks for you guys listening from the beginning until end. And thank you and bye, and see you on next chapter.